Yeah, thank you. We do this approximately 3 times per semester. This semester there'll be 2, another one in November. And in fact, physics of behavior was, I think, the first such symposium that our center sponsored. I mean, there had been things before that, but that was the first official one. And we've come back to revisit the topic every couple of years. And it's exciting because there's been extraordinary progress. So I will remind you that not so long ago, if you wanted to study human or animal behavior, the way you did it was to constrain the behavior dramatically. So our understanding of perception, for example, was based on asking people to choose between a very small set of alternatives. Did you see it or didn't you? Was it coming from your right? Was the sound coming from the right or the left, and so on? And in the meantime, our neuroscience colleagues had this explosion of techniques for studying cells, circuits, molecules in the brain. And so there was this enormous mismatch circa 2000. And in the intervening decades, there's really been, I think, a revolution in our understanding of behavior, or at least in our ability to characterize it. And that is already now in a second generation, which you'll see represented today. So the idea is to try and characterize more naturalistic behaviors, more quantitatively and more completely. And there were first tools that were tuned to the particular examples, and as you'll see in Talmo's talk now, tools that are much more general purpose. So what's happening is that you have an explosion of our ability to characterize behavior in a wide range of animals. I guess today you will see flies, mice, and worms, which, as biologists often joke, are all the animals that exist, and except for us. Will there be any humans?
Oh yeah.
Okay, we'll get humans too. Just another model organism. And anyhow, these data raise all sorts of interesting theoretical questions. There are practical issues about how to analyze them, but also conceptual problems that are just beginning to be unearthed. So I think we're in for a great day. And it's a pleasure to start with Talmo Pereira, who's coming to us from the Salk Institute, but is someone whom we know reasonably well. It's nice to have you back, as I teased you as a grown-up. So please. And I guess you should say this, but I'm going to say this on your behalf, particularly since the group is small, interruptions are encouraged, and it's your job to manage them. If they get out of hand, I'll step in. But
Sure. Okay. Thanks, Bill. Just I guess in terms of timing, I want to make sure I don't cut into other people's times or the break.
So the break is 11 minutes.
Okay, great. Well, it's plenty of time, but please do feel free to interrupt. Okay, so thanks, Bill, for that kind of introduction. I'm Talmo Pereira. I am a Salk Fellow and Principal Investigator at the Salk Institute for Biological Studies. But as Bill is alluding to, I received my PhD in neuroscience in Princeton just last year with Mala Murthy and Josh Shavitz, who was part of the center. And it's lovely to be back here. Today, I'll be telling a little bit about some of the work that I did during my PhD in quantifying behavior using deep learning, and how my lab is now taking some of those core technologies and applying them to a variety of new kind of application areas, and describing some of the new kinds of both analyses and application areas that this enables. So with no further ado, let's get into it. All right, so behavior in a very broad sense can be thought of as movement, right? One of the core reasons that all forms of life evolved the ability to move is to be able to survive and adaptively navigate or adjust to a dynamic environment, whether that's a plant growing through soil, a fruit fly scratching its head, or a giraffe walking through the savanna. All these forms of life evolved the ability to move. And if we want to understand them in a very broad sense, it would be beneficial to have the ability to quantify all these types of motions. In other words, to extract the core and essential elements that encompass all the degrees of freedom that underlie all these different types of movements across all forms of life. So how do we do this? Well, coming at this from the neuroscience perspective, but later I'll show you how this generalizes. We can think of the approaches for quantifying or capturing the dynamics that underlie movement or postural dynamics across different levels of precision. Starting at the coarsest level, that of sensory tracking. This is what's been possible using classical methods for a while now. It suffices to, for example, characterize the dynamics that underlie some behaviors like navigation, right? Something that describes where you are in space. The center of mass, our point animal, is often a sufficient simplification of all its movements to describe pretty complex types of navigation-related behaviors. But if we want to characterize other types of behaviors, such as this fruit fly grooming its head, what we're going to need to do is to move down a level to what we call single animal pose estimation, wherein now we not only need to track the center of mass of the animal, which, as you can see in this example, is not really changing at all, but we also need to be able to track the individual body parts that are actuatable in this animal's morphology. And at an even finer level, if we want to understand the complex dynamics of the interactions between multiple animals, in the case of social behavior, not only do we want to be able to track the location of all the individual body parts of our animals, but also multiple of these animals interacting in potentially complex ways. So one of the motivating questions then for the work that we do in my lab is, how can we use deep learning to facilitate our the ability to quantify behavior across these different levels and across different application areas? So what I'll be telling you about today will be initially some of the early work that we did in single animal pose estimation. I'll introduce the problem and what we needed to solve in order to address the issues related to doing this at this level of precision. Then some of the newer work that we've done in developing a software framework to enable multi-animal pose tracking, and some of the generalizations that we had to overcome to get down to this level of precision. And then finally, I want to show you a series of vignettes illustrating the types of applications that we've been working with our collaborators on, and the kinds of new types of science and analyses that this technology enables. All right, so let's get into it, starting with single animal pose estimation, or how can we recover the locations of individual body parts in individual animals. So as I mentioned, the problem of single animal pose estimation is essentially that of going from videos that might look like this to the location or coordinates of individual landmarks along the morphology of our animals that might look like this. And to be clear, when I show you some of these videos, what we're doing is we're defining each individual body part as a distinct or unique type of landmark, such as the head, the nose, the base of the tail, and so forth. So our goal is going to be to attempt to regress these coordinates given just the raw image. So how do we do this? Well, the approaches that, yep. Go for it.
Is there a step in the check that your set of landmarks is sufficient to reconstruct videos? There is a variability that you're not capturing.
Yeah, that's a great question, Bill. I'm glad you asked that. So for what I'm going to be showing you today, it is largely done in this supervised context where the experimenter or practitioner is defining the set of landmarks that cover the span of the body. There is some work that attempts to essentially discover what the relevant landmarks are, subject to some constraints like ensuring that you can reconstruct the video or perhaps like the motion between frames and so forth. But we are working on some of those techniques. But for what I'm going to be showing you today, and for some of the initial work, we're going to rely on our biologist colleagues to determine what are the relevant landmark types. But that's a good question. We can come back to that discussion a little later. All right. So how can we solve this problem of pose estimation using deep learning? Well, some of these approaches were first developed to track human movement. And these essentially started being described around 2016, where we went from a regime where it was incredibly difficult to achieve reliable motion capture to then being able to leverage deep learning to get what is not totally perfect tracking, but certainly very, very impressive, considering that this is a moving and non-static, non-standard camera angle. The dancer is wearing black clothing against a black background, which, if you've ever tried to use classical image processing techniques, this is a bit of a nightmare. And yet we're still able to get pretty reasonable results. So when we first saw this, we realized that this is probably going to be the right technology for solving this problem in animals. So how does it work? So we start by defining a big convolutional neural network. And if you're not familiar with deep learning, that's okay. Just think of this as a large function approximator that takes an image as input and is going to produce as output a set of what we call confidence maps. Confidence maps were one of the essential advances that enabled deep learning to be able to solve this problem in a really tractable fashion. And essentially what they are a spatial probabilistic representation of the location of each landmark or body part. Think of these as a multi-channel image where each channel is a heat map that corresponds to a unique body part. And so if our network is able to now predict these heat maps, these confidence maps, given the original image, we can then recover the coordinates that make up the pose by finding the pixels that have the highest probability in each for each landmark type. Yeah. That's a good question. So in the single instance case, right, where let me repeat the question. So what about the connections between them, right? I'm showing these as stick figures, or you can think of them as graphs where the individual landmarks are nodes and the edges are the connections between them. In the single instance setting, there is no notion of the skeleton. That is done purely for visualization. And in part, this is because if you know that there exists precisely one person, there's going to be exactly one neck, one elbow, one left wrist, and so forth. So we don't really need to consider that part of it, but we'll get back to that when we get to the multi-instance setting. All right. So given this general construction, it seems like there's no reason why we can't swap in an image of a fly for the image of a human. So what was the impediment from just directly applying these methods? Well, while for humans there exists these massive labeled data sets that have hundreds of thousands of images and millions of individual humans that were annotated through large expense over many years by large industry labs through approaches like crowdsourcing. For any new animal, any new lab animal, any new imaging condition, experimental setting, or number of even different morphologies or number of body parts that you want to track, there are going to be precisely 0 new labeled images, certainly when we started. And so this is what we call the labeled data problem. And to address this gap, we developed a method called LEAP. LEAP stands for Leap Estimates Animal Pose. It's a cute recursive acronym that was coined by Diego Alderando, who was my undergraduate mentee when he was at Princeton. And LEAP works essentially the same way that the human pose estimation models do. It takes in a raw image, feeds this into a convolutional neural network that then learns to output a set of confidence maps from which we can estimate the locations of the individual body parts. Now, the key difference is that we reason that if our convolutional neural network has fewer parameters, that is, if we design a neural network architecture that's much more lightweight, it's also going to require a lot fewer labels, right? Thereby helping to mitigate this labeled data problem. All right, and so fewer does not mean 0. So we designed it in the architecture and we tried it out. And what we found was that even with as few as 10 labeled images, we were able to train these neural networks to get to pretty reasonable accuracies, where here the outer circle corresponds to where 90% of our predictions fall relative to the ground truth. For some body parts, it was doing reasonably well. For many body parts, not quite there. But what we found was that even when we got to only a couple of hundred labeled images, we were already getting not only plateauing accuracy, but essentially perfect accuracy with respect to the ground truth. And so that was terrific because it demonstrated to us that we didn't need to hit that hundreds of thousands of labeled frames data set size that had been previously demonstrated for humans. But when we evaluated this method, we found that not only did it require fewer labels, but by virtue of being lightweight, it also exhibited a really fast training performance. What that means is that on a single conventional desktop GPU, we were able to get to basically perfect accuracy on the order of just a few minutes. Again, owing to this lightweight network architecture. Not only did we train very quickly, we were also able to predict on new frames extremely quickly. And as a result, this enabled what we call a human-in-the-loop training or labeling protocol, where essentially we train a neural network very quickly on very few images, predict on unlabeled frames, and then import them in to correct only the mistakes that the network has made. And what we found was that using this approach, we're able to reduce the time that it takes to label every new image down from around 2 minutes to just a few seconds by the time that we get to plateauing accuracy. Yeah. Yeah, that's a great question. So why even have these large, many parameter, less lightweight neural networks in the first place? So definitely in the human case, right, they're attempting to be able to handle and generalize across a much wider domain with many more complex types of imaging conditions than we typically find in the lab. So really part of the intuition here is that we're scaling down the neural network to match the variability that we see in the lab, in lab domain data in the first place. But there is still room for large neural networks, in particular in terms of developing models with large representational capacity. It's just that in our experience, we've not found that to be truly necessary, because a reasonable workaround when you're doing this for scientific applications is to just train these specialist models that can do really well on a particular data set, as long as it takes relatively little time. And while we could develop really generalizable models, and we are pushing a little bit along that direction, general purpose models are never going to be as accurate as the precise models that work really well on a given data set. So at best, we'd still want to fine tune these large scale models. And it begs the question as to why would you want to start from there in the first place, and how much time does it really save you over using a lighter weight model? Mm-hmm. Yeah, that's right. And we'll talk a little bit more about a more principal analysis that we did in our follow-up work to understand what aspects of neural network architecture design really contributes to this capability. So we'll come back to that point. Okay, so we found that LEAP worked well, generalized across a whole bunch of different conditions, even ones that are a little bit more complex, as in don't have just like a static or a simple background. And we'll, I guess, leave it at that for the moment. I want to mention that concurrently with our work, another approach was developed. It's well known in neuroscience now, called Deep Lab Cut, which uses a slightly different approach. In their approach, still it has the same setup. We have an image of an animal that goes in as input, and a set of confidence maps that get produced as output. But the key difference was in how they handle this labeled data problem, where rather than scaling the neural network down to fit the size of the problem, they instead employ a very common technique in computer vision and deep learning called transfer learning. And here, the intuition is that you start off with a larger neural network that has been trained on a larger data set, for example, something like ImageNet, on a task like categorizing classes of images, or classifying the images into categories rather. And the intuition for why this approach works is that if you have a network that is able to just generally detect a very large and generic basis set of image features, that with only just a few labeled images, you're able to then fine tune, or starting off from that point, modify further train the network to then optimize against solving the animal pose estimation problem on your particular data set. And so this is a bit of a contrasting approach in that you are using a larger neural network, but the premise is that you're starting off somewhere much more favorable in the optimization landscape of the model, rather than constraining the landscape in the first place. So this worked well, and between our two approaches, the field certainly experienced a very rapid growth for all sorts of new innovations that enabled all sorts of things, such as 3D pose estimation and pose estimation with particular experimental constraints. And should say that we reviewed this in 2020, which is now getting a little bit out of date given the pace of this field. But we'll leave it there for now. And I want to switch gears slightly and start talking a little bit about some of our newer work on multi-animal pose tracking, or, yep. That's a great question. So when I showed you a few different videos, for example, of these example cases, these are all different networks, right? We train one for each data set type with the idea in mind that we can develop a specialist network that can begin to track any new type of data in the order of minutes, with on the order of tens to hundreds of labeled frames. I'll also mention that, in this case where we have different sets of landmark types, we're always going to have to train a different model anyhow, because the output layers will have a different shape. We're regressing against different landmark types, even if they could handle different types of data. There was another question. Yeah. Yes. Mm-hmm. Right. Yeah. So what? Yeah. So the question being, what about depth? Can this method handle depth? Yes. Essentially, we can treat a depth image and render it out as a 2D image and just treat that as a generic input. And plenty of folks have done that. I think that was actually part of our original tutorial for LEAP. Then that allows you to get a semi 3D estimate from a single view. But I'll show you a little bit later a quick example of where we're pushing with 3D with regard to using multiple views. Yeah. Mm-hmm. 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 Yeah. It depends on the specific example, right? Yeah. So we do have a notion of visibility, right? Where in our original approach, we kind of more so relied on the strategy of what's called hallucination, where essentially you just, when you're annotating the images, you position the ground truth location of the landmark at your best guess of where it might be. But what we later found was that we can actually get much more reliable estimates by actually explicitly marking some body parts as not visible or missing. And essentially what the network will do is going to be to learn to output a confidence map that is either, somewhere between 0 and 1, and essentially it decreases the probability of its confidence about a particular landmark if it's not visible. And this can actually reduce the noisiness and that you find in the data by quite a bit, because you can just drop all the points that have low confidence and then perhaps interpolate in between if you need continuous data. Yep. Cool. Yeah. Sure. Right. Yeah, that's a good question. I'll say that for this original work, we did not do quite too much of that, other than by breaking down our error rates by body part type. and indeed finding that there is a significant difference in their distributions. What we'll see in a little bit is that we think that one of the core reasons for varying performance pertains to the length scale of the particular anatomical features that we want to track. In the single animal pose estimation case, maybe this is a little bit easier to analyze than it is in the multi-animal case. There we introduce a few more error types, but maybe we can talk a little bit more about that after I show you some of those results. It's a good question though. Yeah. Cool. All right. So single animal pose estimation solved. No problem. Easy peasy. So next we wanted to tackle the case of multi-animal pose tracking. We thought, well, we did the hard part. We can find the individual body parts of a given animal, any animal, given enough data and a handful of GPUs. So it shouldn't be that hard to extend to multiple animals, right? Well, first off, we are interested in social behaviors which is where we find the multi-animal setting, specifically because they are very diverse and complex. Complex in the sense that they have a little bit more complicated structure owing to the relational nature of social behaviors. From an engineering perspective, this is also challenging in that the animals will now be occluding each other quite a lot, meaning we have a lot lower visibility of specific body parts, and it introduces a couple of additional challenges that we didn't have in the single animal setting. Namely, we identified two core problems that we had to solve in generalizing to the multi-animal setting. While it was easy enough to extend the task of localizing body parts to handle multiple instances or multiple copies of each body part type. For instance, here we have multiple heads, multiple tails, and so forth. We're next faced with the problem of what we call part grouping. That is to say, given a set of landmark detections, how do we associate them to each other such that they form sets that only belong to a given animal, right? It's not always the case that the ones that are most closest to each other belong to the same animal. This is especially not the case when the animals are closely interacting, which is really the core object of study of this modality in the first place. So we had to solve this problem. The second problem we had to address is that even assuming that you have not only detected or localized all the landmarks within an image, group them together such that they belong to the same animal. Now, across multiple frames, we need to know which ones belong to which. That is to say, ensure that a set of detections, which may have moved, which may have noise, belong to the same animal, or in other words, can be assigned to the same identity class. And so to solve these additional problems and to generalize what we were able to do with LEAP, we developed a new software framework called SLEAP, or Social Leap Estimate Animal Poses, a cute double recursive acronym. SLEAP was designed from the ground up as an open source software in Python that is aimed at the non-technical practitioner to take them all the way through the entire workflow of having just some raw unlabeled videos through to annotation, configuring and orchestrating the neural networks, to applying them on new data and pulling them back in for manual inspection for correctness. Suffice it to say, SLEAP essentially solves this core problem of going from raw images of the animals like you see on the left to the locations and groupings and tracking of individual body parts for any number of landmark types, a number of animals or type of morphology. And this would not have been possible without the hard work of a series of very dedicated software engineers who have now been working on SLEAP for about 3 years. And our user base is growing in large part, we believe, due to the effort that we put into the usability components of this software. And I'll mention a little bit more about that in just a moment. But here you see a little sampling of the types of data that we developed SLEAP for and evaluated against in our recent paper. And I encourage anyone who's studying or has similar types of videos that they like to quantify to get some SLEAP at sleep.ai. So what went into making sleep a useful software framework? In large part, we spent quite a lot of work in not only developing out individual submodules that implement all the functionality in sleep, but also in organizing it and ensuring that it is extremely accessible, even with no coding experience whatsoever. In fact, you can work through the entire workflow of starting off with raw data to getting a set of coordinates that are tracked and that you can use for downstream analyses without doing any coding at all. And again, as I mentioned, this is a fairly large and mature software framework, which is enabled by the adopting industry grade best practices for software engineering, especially in a production level environment, and leveraging a set of technologies, which, as I'll show you in a moment, enable all sorts of goodies that we didn't even think were going to be possible when we started. So suffice it to say that when we tested out sleep against a variety of existing software frameworks in the single animal pose estimation case, which is still one of the capabilities in sleep, not only were we getting the same or improved accuracy, but much faster speeds when it comes to predicting on new data. And this extended to the multi-animal setting, both in terms of how fast they are to train, as well as how fast they are to predict on new data, again, relative to existing software frameworks. But I introduced the core problems a moment ago that we had to solve. And one of the lessons that we learned in developing sleep and making it available to a progressively wider community is that there is just a very large space and variability of problems and application areas in which you could potentially use sleep. And we realized that a single approach is never going to fit all possible cases. And so starting with the part grouping problem, let me tell you about how we ended up solving this problem in a couple different ways, and when one is going to be better than the other. So starting from the approach that we call the top-down, here the algorithm, the idea is to first find all of our individual animals and then locate their body parts. And the way that this works is by taking an image, feeding this into a first stage neural network that learns to detect a set of anchor points. You can think of these as either the centroids or a particular body part that's highly visible on each animal. We use then this detection to crop a bounding box that's centered on each animal and feed those into a second stage neural network, which then learns to estimate the poses just for the centered instance, just for the centered animal. And therefore, even if there are other animals or other of the same body parts present in the periphery of the image. And this allowed us to solve the part grouping problem implicitly by essentially imposing a spatial prior through this centered crop. And yep. That's a good question. Training time scales fairly modestly, but inference time will scale a little bit differently. And I'll come back to that in just a moment. It's a good question. Cool. So in the second approach, we developed to tackle the part grouping problem, which we call bottom up. We first detect all the body parts and then group them into animals. And we do this by feeding in an image into a single neural network, which then learns to essentially locate all the individual body parts by predicting a set of multi-peak confidence maps. And we learn to representation of their connectivity. And the way that we do this is by defining a directed graph that spans all of the nodes that describes the skeleton of our animal, and then asking our neural network to predict a vector field that's pointing from each body part to each other body part that it's connected to. And this is a representation called part affinity fields, which was also originally developed for human pose estimation, and that we've adapted and modified slightly to work in the animal domain. Combining these two representations, we're able to derive a set of scores which is essentially the alignment between the spatial vector that connects two body parts and the line integral of the vector fields in between them for our representation of connectivity. And you can see this score, this alignment score, represented in the width and transparency of the arrows there. And so we can consider then every candidate set of body parts by traversing through the skeleton tree and connecting and finding the optimal matching for each set of body parts, essentially solving a k-partite graph partition problem. Essentially, what it allows us to do is to group those detections in explicitly by modeling the connectivity between them rather than imposing some spatial prior on them. Cool. Yep. This is still being done frame by frame. Yep.
The other question, I can imagine settings in which individuals in an algorithm will use.
Mm-hmm.
And as you pointed out, if you're solving different
Mm-hmm.
one at some level it should be really easy to detect.
Yeah. Yeah, absolutely, right. So how do we do with like variable number of potential detections or potential edges that we need to assemble, right? Yeah, and that can come up if the animals go, leave the field of view and come back, so we have different numbers, or if we just fail to detect a particular body part because it's occluded, or we have erroneous, additional detections from our neural network, just not doing a great job. This is all handled by making some simplifying assumptions, such as that the directed graph that spans the nodes underlying the skeleton must form a directed tree, essentially. And if this is true, there's actually some nice theoretical guarantees that even solving this greedily, edge by edge, still gets us a globally optimal solution. And it's still able to handle the notion of having a variable number, even at each individual edge, because we treat them all as like local bipartite matching problems. And this has some limitations, but it does ensure optimality. Okay, and so we implement these two approaches for flexibility. And when we compare them across a whole bunch of different data sets, we found that not only do they vary in terms of performance based on the data set, in terms of accuracy, but also in terms of speed. And one of the clear results that we got that that was expected just from the theory and understanding how these algorithms scale was that the scaling in the bottom-up approach is essentially flat with number of animals. And largely this is because we do a single forward pass through our neural network, regardless of the number of animals. And the graph parsing portion of the algorithm is relatively fast. Whereas in the top-down approach, because we now must do a forward pass in the second stage of the neural network for each individual animal that we have, now this is going to scale almost linearly with the number of animals. So at particular regimes, there's going to be a preference towards one or the other. Yeah. Yeah, so on the x-axis, we have just the speed, or how many images per second we can process when we're predicting on new data. And on the y-axis, we just have the accuracy or how well we're approximating these poses. And underneath the map, this mean average precision metric, we actually folding in a few different sources of structural error, as I alluded to earlier, including accounting for visibility, for relative scale, and a couple of other components. Yep. Right, correct. The top-down approach has no explicit notion of the connectivity of the body parts. So in that one, the definition of the graph that underlies the skeleton doesn't even matter. We just mostly use it for visualization. Yeah. So one is the theoretical maximum here, and this is a data set wide value that's scaled to, like I said, to incorporate several different sources of structural error, such as, for example, normalizing to the uncertainty of human annotation error, right? So we actually use some constants that are derived from crowdsourced data to estimate what the human annotator precision is going to be for a typical like easy body part. So it's going to be incredibly difficult to get to exactly one, but these are all in the same units at least. Right. A little bit more complicated than precisely that. We use another metric called the object key point similarity, which explicitly parameterizes your localization error by essentially treating the error variability as a Gaussian centered around the population mean when you ask a bunch of people to click on the same point, right? We can account for that variability, and essentially what you're getting is something's kind of scaled to that amount. But for the mean average precision, we're actually incorporating a couple of other sources of error, such as false negatives, right? If you fail to detect a point entirely, right, there is no error distance there really. So we kind of fold that into this average precision metric. Yep. It shouldn't change at all with the kind of background you have. That there is an architectural consideration there, but let me keep going for a second, and we'll get to a story that touches on that. All right, so the second big problem we have to solve and moving to the multi-animal setting was that of identity assignment, or keeping track of which set of detections is which, and ensuring that they belong to the same animal across multiple subsequent frames. And again, we went for flexibility in the approaches here by developing two different techniques for solving this problem. The first one is what we call temporal association, in which essentially we apply optical flow, essentially a technique that estimates the movement or displacement of image patches, and use this to predict where animals are going to be in a new frame even before doing the pose estimation. Then we'll run the pose estimation on a new frame and align that with where we expected the animal to be, given our estimate of the displacement of its individual body parts. And this flow shift approach works pretty well, but it's not totally perfect, right? It's gonna, even if we get down to just a few, identity switches per 100,000 frames, which is what we were getting in our benchmarks, getting it wrong once means that it's going to be wrong for the rest of the time, right? Animal 1 is going to be animal 2. And that's not going to work for your analyses, even though there's only a single error event. So this notion of error propagation is kind of incompatible with a variety of different settings, which motivated us to develop an alternative approach for solving the identity problem, which we call direct classification. And in this approach, we don't leverage time at all. Instead, rely on the notion that animals have a distinct enough appearance that we can classify them out and assign a probability that each new detection belongs to a particular animal class or identity. Here, denoted by the bars above each box, associated with the male versus the female fly. And we do this differentiably and in tandem with our pose estimation. And we developed a couple new variants of our neural networks that learn to essentially solve this classification problem at the same time that they're doing the tracking. And this is important because it allows us to basically solve the multi-animal pose tracking problem completely end to end. And I'll show you a little bit more about what that enables in just a moment. But first, just to give you a sense for the difference between these approaches, the most common setting in which we find that we get identity errors is when there are also pose estimation errors, meaning some body parts are too blurry, they go missing, or we just misassign them. In the temporal approach, whenever a situation like this happens, we get it wrong once and it remains wrong. Whereas in the appearance-based approach, which doesn't depend on time, this is going to resolve itself automatically in the next frame, or as soon as it's unambiguous which identity is which. And this allowed us to work with a particularly challenging data set here of a set of gerbils that are continuously monitored. And what that means is that they're recorded at a high frame rate for upwards of weeks at a time. So even with an incredibly low error rate, it would be intractable to manually proofread these because it would require going through all these frames, or at least a subset of them manually. And so here what you can see is that in this one of the cutest data sets I think we had in our paper, these gerbils really like to essentially get into these huddles or these cuddle piles, which incredibly cute, but very challenging to to handle on the algorithmic side, because now we have to deal with the fact that they're completely on top of each other, their body parts are intermingled, and you can see that when they are on top of each other, indeed our accuracy does suffer, but as soon as they split up again, the identities resolve themselves, and we're able to essentially still process this data set and get reliable tracking for the vast majority of the time without having to do any manual proofreading or interventions. Right. So let's see. As I mentioned, sleep is a complex, is a developed machine learning system, meaning it's a large software framework that has many different modules associated with them. And optimizing each one of them individually was an entire sub-project in of itself. And I just want to tell you one little story about one of these components before we move on to some cool examples. Yep. Yeah, and in fact, some of our collaborators have done precisely that by leveraging like QR-like codes that you can place on the animals and use that as a parallel signal, either that you apply post hoc or in tandem. That is something that I think at the moment is best done post hoc, but we certainly are exploring methods to do precisely this. We've been getting some really fun new results in both differentiable, like temporal-based tracking, and differentiable and unsupervised, or rather self-supervised discovery of identity appearance vectors. We're definitely going there. Okay, let me just speed through this little story here real quick. This is the only time you're going to see an equation, so I don't want to be remiss. So we talked a little bit about architectures, right, and how a lot of the innovations that we push was in engineering new neural network architectures that are lightweight and scaled down to match the size of the problem that we were encountering. But it is beneficial for us to understand what aspects of these neural networks precisely are guiding principles when designing these architectures, right? We can treat them as generic black boxes and use these general purpose models, but again, we always find that understanding precisely what the networks are doing and how to structure them always gives us better, or gives us more gain. So we developed this very generic encoder-decoder architecture that is essentially made up of a few set of blocks, of convolutional blocks, sampling blocks, and a couple of other blocks that are just essentially motifs of neural network layers in their topology that can be varied independently and that translate into different implementations of the actual precise architecture. So think of it as a more modular or hybrid architecture, or meta architecture, if you will. And one of the guiding principles that we discovered had a tremendous impact on performance is what we call the receptive field size. And for the receptive field size, there's a closed form equation that relates this property to the specific architecture, meaning given an architecture, you can predict what its maximum theoretical receptive field size is going to be. And this is a property that is essential because it determines the length scale over which our neural networks are able to reason over image evidence. And we intuited that because our data sets had different resolutions tied to the physical size of the underlying anatomy of each animal, that there was also going to be a different optimal receptive field size according to the scale of both the animal and its features. So what we did was we varied the neural network architecture specifically to modify the receptive field size. And we find that even though it's a high dimensional hyperparameter space of neural network architectures, varying this single parameter could reliably increase our accuracy up to what was like our overall best performance on each data set, demonstrating that modifying this particular property of neural networks was sufficient to get us to optimal performance, and indeed is a single hyperparameter that can be tuned that can get you to confer your neural networks the ability to solve the task as appropriate for your data set. Again, we find that this works quite well across species and conditions, but crucially, I just want to do a little callback here to this alternative approach, which is of leveraging what's called transfer learning. And so we wanted to see how this notion of like a principle-guided architecture design stacked up against the more commonly employed approach of transfer learning, where we use a pre-trained part of the network that is much larger and has lots of features already built in, and then just fine-tune it on new data. And what we find is that for a variety of leading neural network architectures, indeed, while transfer learning does improve the accuracy over training the same models totally from scratch or from random weights, that they at best do as well as our principle-guided lightweight architectures. And we tested this out against a very wide range of different neural network architectures, and find that while they at best will give us the same accuracy as our custom designed neural networks, they'll always be uniformly much slower because, again, they're much larger, complex, and intended for more general purpose domains. Yep. Yeah, so these are essentially just two parts of a neural network, where the encoder takes an image as input and will repeatedly apply convolutions and downsampling to increase the receptive field size, but at the cost of decreasing the resolution. The decoder will take a small resolution but high dimensional set of features encoded at each pixel block, and then upsample it back to be closer to the original image resolution, fusing in information from the appropriate scale level in the encoder branch. Yeah, it gets into some nitty-gritty there of neural network architecture design, but suffice it to say that it essentially learns to detect image features across multiple scales. Cool. So this validated our general approach that, yes, we can do much better by just smartly engineering our neural networks to fit the size of the problem and particular high-level properties that we find in each data set. But in addition to being faster theoretically, there's just fewer computations in these smaller models. We also put a lot of work in the implementation. So in the engineering design of these models, in particular in the ones that do end-to-end pose estimation, we want to see how fast, fast really was. And in the offline setting, where our goal is to maximize throughput, we were able to get way past real-time performance on each one of our data sets. But what we're really interested in was whether these improvements in performance, they're nice, are they nice to have, which do more things faster, or can they enable something entirely new, like a new experimental application paradigm? And this is where we find that that these improvements in performance not only increase our throughput, but decrease our latency. So how fast we can predict on new images. So what you're seeing on the right there is how long it takes for us to get an image from a camera and get back the location of 26 body parts, group them such that they belong to the same animal, and classify them into which identity they belong to in just around 12 milliseconds. And that's fast enough to be at the scale of being able to manipulate and intervene in biological processes. And we were pretty happy with that until we connected with some folks at NVIDIA who helped us implement an alternative inference engine using their own library, which is a lot more aware of hardware specific accelerations. And just by dropping in and swapping out this software framework, we were able to essentially triple these numbers, both in terms of throughput and in terms of inference latency, meaning that now we're going from this 1024 by 1024 pixel image, super high resolution, meaning we're not sacrificing accuracy at all in just 3.5 milliseconds. And I didn't even think that was possible, because you're moving a lot of data to the GPU and coming back. But we wanted to do a little proof of principle to demonstrate what this could enable. And this is what I call social mind control, in which essentially by experimental design, we we put a female fruit fly who is sexually receptive in a courtship setting with a male fly. We can track both of them and detect when the male fly is approaching, and in real time, force her to reject his courtship advance in a situation which she would not have otherwise. So put in other words, we can now in real time detect the social behavior of one animal, intervene in their typical courtship ritual, and trigger a social response behavior in another animal by activating some neurons in its brain. Demonstrating as a proof of principle the kinds of new applications or types of experiments that you can do, given that you've cleared the engineering hurdles of being able to do this quickly enough. Cool. So I'll just end this section with just a couple of demonstrations of where we're going next. In one very natural extension of this is to extend this to 3 dimensions. So while this works just fine in 2 dimensions, especially in the context of social behavior, we have additional challenges when we add this one extra dimension. And we can get around this by having multiple cameras that are looking at the same, synchronized to each other and calibrated. And what we're now working on are methods that can leverage multi-view information to better do the pose tracking when there's information that's missing from each individual view. And then, given that we have reliable 3D pose tracking, we further want to now take this and use it to power simulations of our animals' behavior by essentially registering the 3D motion capture to biomechanically realistic models of the animal's morphologies, and essentially convert the 3D locations of the landmarks into a set of essentially torques that can get applied to each muscle and obey the laws of physics and the kinematics of each animal's body. And we believe that this is going to unlock a totally new way to analyze behavior by modeling the specific generative processes that give rise to it, namely motor control of an embodied agent. Okay, that's the end of our section on sleep, and I want to finish off. I know I have like 30 minutes or so, right? I don't want to cut into a break too much. Just a small series of vignettes, but I'll take a couple of questions on sleep now, and we can come back to it at the end. Yeah. Yeah, so this is pretty, yeah, this is, yeah, so yeah, this is a very much intended to be a teaser, and happy to tell you a lot more about it offline. But yes, our goal is to be able to, once we have these guys instantiated in simulation, then to leverage this as a scaffold for then developing models that can learn to imitate the same movements of real animals, and thereby learning the same kinds of organization and computations and algorithms that underlie behavioral programs, all the way down to the level of, millisecond scale kinematics. Sure. Yeah, I think, our view is that if we have the capability to, map motion capture onto a biomechanically realistic model, then the world's your oyster. It kind of just serves as a platform for whatever level and flavor of modeling you want to do that gives rise to those behaviors by essentially giving you a physics-equipped generative system for studying it. Yeah. Okay, so let me show you some of the cool stuff we've been doing with this before we run out of time. And this is going to entail how we've been working with our collaborators to create new kinds of applications and use cases for this kind of technology. A very natural one that we started developing as soon as I joined the Salk Institute, which we say is where cures begin, was to point this technology to solve the problem of disease phenotyping, right? So we did all this work to make sleep extremely not only accurate, but fast, right? Fast in terms of latency, also fast in terms of scalability. So we thought that, okay, a very natural application of this is to scale it up. So we got a ton of GPUs, and we started to develop this system that we intend to scale up to our entire behavioral testing core, which essentially involves placing cameras on every home cage in every behavioral setup that's present in this core facility, allowing biologists to just bring in their animal, which might be a model of a particular disease, such as cancer or neurodegenerative disorders, and get out all the analytics that we can on top of it, and to do this at scale without requiring manual interventions, requiring dedicated staff to go and score or move animals between cages and so forth. In here, you can see what that kind of look at that level of data kind of looks like. Where here we have 300 hours of mouse behaving in a single home cage, totally unattended, and obscured here. Going into this little animation is something on the order of a few 100 million data points that altogether were collected in a totally autonomous fashion, and that doesn't require any manual intervention. But we still want to get some type of analytics out of this. And so what we're now doing is building a set of tools and analysis frameworks that allow us to derive more meaningful insights, such as from the motion capture data, such as are the animals interacting with one another in a very general sense? Are they perhaps interacting with the feeder where they can actually get food? That's going to be an important readout, for example, in one of the pilot studies that we're doing on a pancreatic cancer model, which affects their metabolism and subsequently affects the structure of their feeding behavior. So we can put in a mouse that has pancreatic cancer, another mouse that has pancreatic cancer but has undergone a particular form of chemo, and observe whether that recovers the natural structure of their feeding behavior as a readout that is scalable and that can be later confirmed through experimental means. So that was pretty exciting in that, it's kind of allowing all sorts of new applications, but we're always curious about new approaches for extracting these types of insights. And through interesting, very interdisciplinary collaboration, we began to connect, largely through collaboration with my colleague Valerie Williams at OSU, who's a legendary dance professor, new ways to leverage motion capture to describe movement. And so we recently learned about, or I recently learned about one system for describing movement called Laban notation, which was originally developed by Rudolf von Laban in the early 20th century, where Rudolf was the classic industrialist, and his goal was to maximize the throughput of his factories, and to ensure that his factory workers were moving as efficiently as possible as they could throughout different assembly lines and industrial processes. He came up with an entire symbolic system that describes the precise movement of each individual body part of somebody navigating in a space to ensure that he could choreograph his workers' movements to maximize the efficiency of his assembly processes. And whilst not so much used for those applications today, it is now been widely adopted in the digital humanities as a way to essentially transcribe or digitize things like complex dance sequences. Essentially think of it as a symbolic or sheet music equivalent for human movement, right? And so I thought this was super neat. We always think about how to describe discrete structure or other types of structure and behavior. So it was nice to see that, in a field where the behaviors and movements are a lot more complex and certainly have different needs, they've come up with a pretty sophisticated system for describing all the variability that you see in human movement. And so I asked Valerie to see if this could work with animals. And I just gave her a video of a fly walking around, and just by adding a couple more columns to account for having additional legs, she was able to transcribe out this little short clip of a fruit fly navigating this space into Laban notation, demonstrating the generalizability of this type of approach for describing movement. And so that was pretty neat, demonstrating how we can definitely learn a lot from these different fields that perhaps haven't had the ability to use machine learning to automate the primary data capture, nonetheless still have really good approaches for describing the underlying structure in it. And I wanted to take this a step further and align this directly to the motion capture and understand how this system for describing movement is able to handle some of the more trickier aspects of describing the structure of behavior, such as continuous variation. And so here you can see that not only does this notation afford a way to describe the movement of each individual body part, it also has these modifiers. You can think of these as essentially ways to vary the execution of a particular movement type. If we just zoom into to this little side section here, you can actually see that we have a series of these movements throughout this choreographed dance that alternate between being strong and direct, where essentially if we crop out the clips in which the dancer is executing this in with a strong quality or a strong and direct quality, initially I couldn't really tell what was really the difference. But if you overlay them and if you really squint here, you'll notice that in the strong case, they're not always exactly synchronized in contrast to the strong and direct case, in which there's a lot lower variability and the fist reaches a slightly lower position. And by aligning this with the motion capture, we can actually quantify that really precisely. You can see that in the strong and direct execution, they do indeed have a lower minimal vertical position, but also the variability is modified by which type of movement that they're doing. Again, agreeing with what the description was in the first place and demonstrating that this symbolic system can align well with our intuition, or rather our more quantitative methods for describing the quality of and structure of movement. So this was a fun one, and we're continuing to collaborate in building some systems that can better bridge between these two formulations. But we're kind of excited to think about new ways that we can leverage what has been done in the humanities to describe behavior in even animals and other biological applications. But that was our initial foray into working with human data, and we recently embarked on a new collaboration with the LA County Museum of Art. This is a collaborative project between my lab and Tom Albright's lab and Sergey Epstein at Salk and the LA County Museum of Art, where our goal here is to essentially apply these same methods to describe how humans navigate in a museum exhibit space. And here our goal is to first digitally reconstruct the exhibit. And we went down there with my postdoc, and we grabbed a depth camera and started essentially scanning out the entire space as well as the individual exhibits. And so here's a little preview of what that digital reconstruction is going to look like. We're not all the way finished yet, so I'll do some alignment, but the goal will be to now have a bunch of cameras that are now set up, and this experiment's already ongoing, calibrate them in the same way that we normally do with our animals, by essentially having a checkerboard that we can use to triangulate the different views. And we're now applying our techniques for tracking movement, but now in humans. And recall that one of the distinguishing features of our approaches has been to design these neural networks that are much more lightweight. And we thought that for this project, we're going to have a really easy time because there exists all these models already trained on hundreds of thousands of labeled human images. But as it turns out, even when we grab one that's state of the art, the tracking's not that great. And one of the reasons for this is just because even though these are still human shapes, and you can see that overall, I guess the rough human anatomy roughly correct, it's likely that in this very large general purpose data set, there aren't a lot of images imaged from the top down at this sort of security camera type of viewpoint, demonstrating that even though you can have a very large model trained on a very large data set, it's not going to generalize to totally unseen views. And yet, even using sleep with very, very few labels, and despite the much increased variability that we have in human data, we're still able to do reasonably better. And again, this is very, very early work, but we're hoping to demonstrate to leverage this also to demonstrate the notion of generalizability in these networks. And you can see here this is super fresh, hot off the presses results that my postdoc sent me yesterday, where we've just started applying our triangulation algorithms to do the 3D pose tracking using multiple views of a person navigating through the exhibit. And this is going to run for roughly a year. If you're ever in the LA area and you want to be part of our data set, stop by LACMA, Conversing in Clay exhibit. And halfway through the exhibit, we're going to leverage the 3D motion capture plus the digital reconstructions to develop generative models that can describe the trajectory of humans navigating through the space as a function of the geometry and configuration of the space, which we're then going to guide a reconfiguration or a modification of the layout halfway through the exhibit runtime, and then we'll estimate how these changes in the architecture of the space modifies people's engagement both with each other, with the exhibits, and the patterning of their movements in this space. They do, yeah. So one thing that was shocking to me as somebody who has never worked with humans before was that there's just no IRB approval required at all for this. Turns out we're not using people's faces or any identifiable information. And as it became pretty obvious after thinking about it, you're already being recorded all the time when you're in a museum, because after all, they do have pieces that are very expensive and hundreds of years old. So you're already under security camera monitoring the entire time that you're there. We just added more cameras in a particular space. Okay, and so for the final vignette here, and then I'll finish up, this was our foray into yet another more creative application domain that totally caught us by surprise, but one of our users reached out and mentioned that they were using sleep for the study of plant motion. And as I alluded to in the introduction, plants, like every other form of life, evolve the ability to move so that they can, better adapt to their environment. So using time-lapse videography, you can see that essentially the problem boils down to exactly what we had with the animals. And while we didn't have to change anything algorithmically, we were able to leverage some of the principles that we learned about what makes neural network architectures better suited to specific data types to then handle and generalize across a variety of different imaging and experimental settings in the plant domain, where here for each one of these videos, even though they're relatively small data sets, we're still able to get away with getting this precision of tracking with for most of these fewer than 10 labeled images. Again, demonstrating the ability of sleep to use these specialist models rather than the generalist approach of something that can track anything. And this coincided nicely with when I was just starting my lab at Salk, which has an initiative called the Harnessing Plants Initiative. This is a multi-lab effort to combat climate change through plant bioengineering. And here the goal is to engineer plants that have deeper roots, more massive plant roots, and roots that produce more of these carbon-rich polymers like suberin, which is essentially the core constituent of cork, like you have in wine bottles. They're super stable, essentially carbon sinks, right? Where if we can genetically engineer plants, ideally crop plants, which have a natural scalability and distribution system, then we're going to be able to take a lot more carbon out of the atmosphere just by planting a different type of plant that pulls more carbon into the soil. And so while Salk has world-class plant geneticists that understand a lot about the developmental programs that govern plant root system architecture growth, what they were lacking was a good way to estimate how a particular genetic modification is going to affect the root system architecture, which can get quite complex. And so using sleep, they've now been able to, and we've worked with them to build some, to essentially apply sleep in appropriate ways, to develop a system to do root system phenotyping that's essentially fully automated, that enables the extraction of all sorts of root traits, including things like the relative angles, number of branch points, and depth of different types of roots. And here you can see a little gallery, such as on this top row, going from few to many lateral roots, and in the bottom row, going from short to long primary roots. We're able to, again, differentiate using sleep. And this is now being used to guide even field trials where they'll screen many different variants of a particular crop plant and try planting them out in the field and seeing if indeed these properties that we're evaluating in the lab will hold and enable improved carbon sequestration. Okay. So that was a series of little vignettes on the applications. And so with that, I just want to thank you guys for listening thanks, Bill, for the invitation, the ability to, and the opportunity to come and share some of the work that we've been doing. And obviously my graduate labs at Princeton, our funding sources, and the terrific folks in my lab who really did all the work that I showed you today. And thank you, and happy to take some questions. Hmm. Yeah. Right. Right. Yes, there is definitely a big challenge, right? As you might imagine. One of our approaches for the multi-instance setting really relies on having a fixed number. Really, most of our approach relies on having a fixed number of landmark types. So when you grow a new, spring new lateral root, that is a new set of morphological features, right? So we have to kind of expand our graph. So the way we've been tackling that is by treating each individual root as the equivalent of like a new animal. And here we have two different animal classes for the primary and the lateral roots. The primary, where in most species we'll only have one, lateral, which can have an arbitrary amount, but we're nonetheless still dealing with the fact that plant roots don't have a skeleton, they don't have joints, right? So they're not quite as rigid as animals, meaning that even keeping the fixed number of landmarks for a single root might still not be the best way to approximate it, since they become essentially piecewise linear. So now we're working on methods that can automatically or dynamically adapt to having different numbers of nodes that describe the curve, to better capture the curvature of individual roots. And that requires some new computer vision and some new theory for actually solving the assignment problem. Yeah, right. Absolutely. Yeah. So volume is a big one. And what we're most immediately doing now is extending this to use pose to guide segmentation. So to our knowledge, this approach of using landmark-based quantification of plant morphology is really the only one that doesn't rely on segmentation. And we've done quite an extensive literature review at this point. And it's just ironic because in the segmentation-based approaches, the very next step that plant biologists normally do to extract insights is to skeletonize it out back into a set of landmarks in the first place. Of course, that's going to be a lot less robust because in segmentation, you can't deal with occlusions quite as well or reason about them. And so we're now trying to essentially bridge the two and get both like the core underlying directed graph that spans the morphology, as well as to be able to regress conditioned on pose what pixels belong to a given root. Yeah. Right. Right. Bad news is. Now what? That's a great. Yeah. Yeah. Right. Exactly right. Yeah. I mean, a lot of my cancer biology colleagues, right, have no even concept of using motion capture for phenotyping. Certainly, if you want to detect if they're feeding, you could just put an IR beam in front of the cage and just detect how many times the beam breaks, which is when they're feeding. You don't need to, give Talmo a bunch of GPUs to do that for you. The part of the argument is that we're able to get a lot more than you could using these traditional methods. And we are building, as I mentioned, new approaches or a set of more specific behavior or phenotype detectors that would otherwise require specialized hardware to do. But importantly, we're able to do it in a freely moving and more naturalistic setting. And hopefully that added complexity adds another dimension where now we're measuring not just one phenotype, but many simultaneously. And we can always go back and measure more phenotypes given the same motion capture as we come up with new descriptions of them. And I'm hoping that this afternoon we'll hear also about some cutting edge methods for detecting arbitrary phenotypes using machine learning and new definitions. But I'll just briefly mention that some of the work that we did that was building on how can we use pose specifically to characterize behavior states in a maybe more unbiased or data-driven manner, leverage the notion that postural dynamics have these discrete signatures, right? Where, for example, here in this fly grooming, we can leverage the notion that there is a periodic aspect to the structure of this behavior. And if we want to be able to then define this as something that we might be able to call in English head grooming, but in an unbiased manner, one thing we can do is to take these postural trajectories, extract spectral features, or essentially compute the spectrogram or the power at each time scale for each body part that they're moving at, and leverage this as a way to describe the unique aspects of this movement, or essentially create a spectral profile or signature that corresponds to this kind of behavior, essentially head grooming, where here in a 1 second period, we have 9 cycles, and indeed we have a peak in our spectrogram at roughly 9 hertz for just the forelegs. So it's an interpretable method for extracting meaningful features. And if we grab all of these guys and apply some dimensionality reduction to it, essentially embedded in a 2D manifold, we then get out all these clusters, which when segmented out, correspond to individual times in which the animal is moving in similar ways. And I'll just show you one small set of examples here, where if we pull an example from this part of this embedding, we see that it corresponds to when the flies are grooming on the left side and the left hind side. If we move slightly up in this space, the next cluster over corresponds to when they're grooming with both legs, again, and they're in their hind region. And then moving further up to the next cluster over, right hind grooming. So essentially discovering what we could have written down a description for, but nonetheless is now just coming out directly from the data by using these methods that do essentially, think of it as density-based clustering on some description of the dynamics of motion capture or pose. That said, right, there is still much room, even just for the things that are a little bit clearer that we know that we definitely want, right? Like, for example, just knowing when the animals are overlapping with each other or interacting with specific parts of the environment. Ultimately, right, the theoretical appeal will be in leveraging the fact that motion capture is much closer to the language that the true generative process of behavior is applied to. That is to say, behavior doesn't output a set of landmark coordinates, nor does it output, an embedding space of spectral profiles. The brain really produces behavior by controlling the body, right? By sending signals out from the brain, out to the periphery, which then trigger muscle contractions that can then execute a series of motor commands that underlie all forms of behavior. So if we can just get to that point, convert from motion capture, positional information, to the language of muscle forces, then we're that much closer to the actual language that's spoken by the brain in terms of generating behavior. And our hypothesis is that this is going to give us much stronger inductive biases in terms of developing models that can discover and reproduce the structure of behavior. Through the language of embodied control, right, to be clear. Yeah. Right. Yeah. Can we find individual differences or time varying differences in the overall structure of their behavior? Absolutely. I think this has been done by several folks in all sorts of different settings, including by my former PhD advisor, Josh Shavitz, and collaboration with Bill, I think, in several forums. What we can do is leverage the unique aspects of pose to ground this in more biologically meaningful units, right? The structure of behavior is changing overall, sure, but by having motion capture, we can attribute that specifically to something that has an anatomical correspondence, which gives us an entry point or an anchor to then digging into the underlying biological mechanisms that might explain the changes there. For example, in our high throughput phenotyping system, one of the diseases that we're targeting here, we're really using two core disease models as our test cases. One is pancreatic cancer, which we know affects things like feeding behavior, but also we have some hypotheses for how it might affect sociality. And the other one being ALS, or amyotrophic lateral sclerosis, which is a neurodegenerative disorder. And there we have even more specific hypotheses for the kinds of changes that are going to occur phenotypically, in particular relating to the loss of motor coordination. And while we know what that's going to look like in terms of overall decrease in movement, we don't know how it's going to affect specific behavioral motifs. So it might be that if we now go in and analyze a mouse that has late stage ALS and look at all the times in which it was grooming or interacting with even its conspecific, that it'll still be doing it, maybe even at the same overall frequency, but perhaps in a less coordinated or more variable fashion. And this is where, again, maybe we can borrow some of the language and systems for describing variability in movement that we learn from example, the humanities. Yeah. Yeah.
I don't know, think much about the mapping from the appearance of the animal posture to the use of the physics. Because like if you watch two people open the door, they're gonna use the same how much difference is there? Is that going to be a problem?
Yeah, absolutely. So I guess let me distill your question down. I guess one aspect is, yeah, could you tell if a mouse is blind just from doing motion capture? I agree with you. It's a bit of overkill potentially, but one way that you could leverage motion capture uniquely is by adding an extra layer, like I'm demonstrating here, which is also quantifying the environment, right? This is also going to be a lot easier once we have our entire pipeline and platform for physics-based simulation to reconstruct the environment such that we could even estimate what the animals are seeing or touching and so forth. Directly estimating the sensory inputs of our animals just given motion capture. But surely for something like blindness, you could hopefully pull that out of the overall statistics of their movement, right? They're probably not going to actively foveate quite as much and so forth. Right. And you mentioned also, right, and the other one being like, how can you capture and account for inter-individual variability, right? Because if you're modeling things all the way down to muscle forces, that's going to be pretty specific. And are we going to get stuck in the weeds of biomechanics, precluding us from understanding the higher level organization or control of behavior? It's entirely possible. It's possible. No, there are some specific aspects of doing this in simulation that I find particularly attractive. So I'll throw this up again just to say that one of the key ideas here is that once we solve, and we think we largely have solved some of the core engineering problems of going from motion capture to doing essentially like all of the inverse kinematics to estimate muscle forces for a biomechanically realistic model in a physics-based simulator, is that then doing control over the same articulated biomechanically realistic model is something that can be achieved using a number of different modeling frameworks, including, and obviously this is our framework of modeling framework of preference, including deep learning, right? Where we can use deep neural networks, which afford us all sorts of flexibilities for doing things like abstracting away motor control and yet still having higher order controllers or high level latents that allow us to capture things like inter-individual variability. And I'm happy to chat a little bit offline about how we're planning to do that precisely. But suffice it to say that assuming that we can get things into this language of biomechanical motor control, then our hypothesis is that that's going to significantly constrain the solution space of possible ways to encode out the algorithms that control behavior. Yeah, and by the way, I'll just mention it's 11:32. I don't want to cut too much in the break, but go for it, Bill.
In this issue of using, there's an experiment which I always like. I don't know if it's actually where what they did was they took people, they used
Yeah. A gradient of.
And then you basically pick up the bacteria levels, and then you see.
Right.
And in one afternoon, you recapitulate
Evolution. Yeah, many. Right. Right.
But the point is that using these tools, right, you can then move that in that.
Correct. Mm-hmm. Yep. Right. Yeah. Mm-hmm. Yeah, and there's some work going on, and for example, Sarah Coker's lab, right, at Princeton, who has been doing some of these more traditional, gene by environment, gene by phenotype type screening. The thing that I would be particularly excited about would be to move some of that kind of simulation of, evolutionary pressures into the in silico domain, which is part of the reason why I'm so motivated to devote so much of my lab's resources to breaking into this whole biomechanical simulation domain, because if we can have a neural network which learns to control the body to even just to imitate the real behavior of a real animal, or just to solve the same tasks that a real animal would have had to evolve the ability to do, we're simulating some of the same pressures that gave rise to the brain in the first place. But importantly, we're constraining it by the output space of the biology, of the biomechanics and the anatomy that these neural programs actually operate on. So you could imagine doing things like having this virtual brain, which again is able to take the same sensory inputs as a real animal, produce the same motor outputs as a real animal, and systematically inactivate different parts of the neural network that controls the animal, and find analogs to real parts of the brain, essentially be able to simulate the kinds of experiments that maybe would take many years to do in a lab in an afternoon. Similarly, you could imagine even broadening this even further and simulating things that we couldn't even do in the lab, like proper evolutionary change, right? Start off with like a class of neural network architectures that could describe a theoretical space of potential neural circuits that could implement and still solve the same, sensory to motor transformations, and evolve that progressively, maybe to add different levels of complexity to the topology, organization, or types of computations that are employed, and systematically see how this will affect the model's capability of reproducing what you see in terms of real behavior. So you could theoretically evolve a brain in silico until it can precisely do what a real brain can. and always be able to check against those constraints. Cool. I think with that, right?
Thanks, Bill. Thanks for the invitation. It's great to be here. I'd also like to thank Talmo for the amazing introduction that he did. It fits quite well with what I'm going to be talking now. So I'll be mostly speaking about, how do you go from this high resolution posture tracking of animals to actually gaining some understanding about the dynamics in, a 3D moving context. So generally speaking, where we're, interested and curious about the diversity and complexity exhibited by animals in the way that they move, and how these movements are controlled by a wide range of biological mechanisms across scales, right? So from the molecular motors that are pushing things around inside cells, to the interaction between cells that drives the muscles, to the coordination of the muscles by the nervous system that ultimately gives rise to how we move. Right? Behavior is also the scale at which evolution really plays a role, right? So animals need to find food, they need to find mates, avoid predators. And so studying behavior opens up new paths for an understanding of a wide range of biological mechanisms across scales, from genes to the brain, even to evolution. Now, this is also interesting from a physicist's point of view, because behavior is clearly an emergent phenomenon. It exhibits multiple scales besides being, obviously far from equilibrium and extremely complex. And so as Talmo was showing you before, these days behavior is not only fascinating to look at, but we can actually start to probe these phenomena experimentally, both in the lab and in the wild. And here I'm just showing you a few examples of recent algorithms that allow you to do this. And they essentially just let you track the position and posture of animals with unprecedented resolution. So we're collecting really large scale data. And this is obviously very exciting for the field of animal behavior, but also opens up new space for new physics or analysis tools that allow us to parse these complex data streams. And so in this talk, I'll mostly be focusing on the behavior of this tiny millimetric roundworm here, which is the C. elegans. And we'll use this as a playground to think about how dynamical systems theory and statistical mechanics can help us in understanding animal behavior across scales. Okay, so this is our little playground, and this is a first look at the data. So these worms are essentially, placed in a Petri dish on an agar surface. So this copper ring here basically just prevents the worms from leaving the plate. And there is a microscope you can see there on that little red box there, which is essentially following the worms around and generating these very high resolution, high contrast images we see here on the left. Right, so C. elegans has been a model organism in biology for a very long time, which means that we know a lot about, how it develops, its genes, its brain. And it has a relatively simple brain, right? It only has 300 neurons, but it's still capable of remarkable behaviors. It still needs to find food and mates. It still needs to avoid predators. It's also capable of learning and adaptation. And so this is kind of a nice playground to study quantitative analysis in behavior instead of a minimal example. Now, long story short, we've known for a while how to sort of identify short-time behavioral motifs from data. So Talmo showed you an example of that. Here's a different one where we take sort of video data and we use a dynamical systems approach to extract these short-time behaviors where, you can see that the worm moves forward, it moves backward, it turns. And on the other hand, at the larger scales, we also know how to characterize the different foraging strategies that the worm does. So you can essentially zoom out and just treat this as a random walker, and you can characterize the properties of this random walk. But now the enigma that we're going to be focusing on today is how do you go basically from this short time scale motif? So how does the worm combine them in time to give rise to changes in the way in which it explores an environment? So in other words, how do we put together this sort of finer scale posture level worm-centric analysis and this larger scale sort of random walks that we observe on the right-hand side? Well, this sort of thing has been done in physics, right? Throughout multiple systems that exhibit, multi-scale unpredictable patterns, physics has shown a tremendous success at finding effective coarse-grained predictive theories from the principled integration of microscopic degrees of freedom, right? Simple example of that is Brownian motion, where by sort of approximating the collisions experienced by a particle through Gaussian white noise, we can predict the larger scale diffusive properties of this particle. Now, in order to do this, we generally need either some understanding of the microscopic dynamics or some notion of symmetries or conservation laws that allow us to write down an effective coarse-grained theory. But in general, and in biology in particular, we lack this level of intuition, right? So what we would like to do here is to try to sort of build such effective coarse-grained theories, but working directly from measurement data in systems for which we lack the same level of intuition. And so in order to do that, what we're going to do here is that we're going to make use of the fact that underlying much of these success stories in physics is often the identification of macroscopic large-scale variables that capture sort of the coarse-grained properties of dynamics and evolve slowly when compared to the microscopic components of the system. And so sort of in the spirit of statistical mechanics, what we'll do is that we will search for slow dynamics in the time evolution of probability distributions or ensembles in a carefully defined state space. So the intuition for this is the following. So imagine that you just have like water flowing down the river, okay? On a very fine scale, the water molecules are really just bouncing around each other erratically on very fast time scales. But if you zoom out and start looking at a larger body of water, you actually see that, you know, it's flowing coherently down the river. So this is essentially what we want to do. We want to, in some sense, integrate this fine scale randomness and extract larger scale structures. And we can do that by essentially zooming out and looking at the time evolution of ensembles. Okay, now so due to the property that the probabilities are conserved, we can write down the evolution of densities in this linear form, where you can think about this L as being, for example, if you have a stationary Gaussian random process, it's just a Fokker-Planck operator, okay? It's an infinite dimensional operator, but it allows us to essentially, you know, subsume the nonlinear dynamics in the full state space into, you know, a linear operator representation, which then allows us to decompose the dynamics into its multiple scales, given here by these eigenvalues. And so we can hope to try to identify sort of slow modes that capture the coarse-grained, larger scale properties of the dynamics, such that then we can treat faster dynamics as effectively as fluctuations. This is kind of like the broader picture. We'll look at a few examples in a second. Now, the issue when we're working directly from data is that we actually don't know whether the variables that we measure are enough to capture sort of the full state of the system, meaning whether these variables are enough to describe the future state of the dynamics, or whether we're missing important degrees of freedom, for example, that might induce non-trivial non-Markovian effects to the evolution of densities, or might preclude, you know, important dynamics that are living in a higher dimensional space that we don't have access to because we're only seeing a subset of the degrees of freedom. And so before we proceed, we kind of have to do some sort of inverse physics and try to learn from this subset of degrees of freedom the full state of the system. So expanding the state so that we get a maximally predictive sort of Markovian state space. Again, I'm going to go to a simple example first, and then we're going to talk about the more general setting. So just imagine that you have a particle in a double wall potential. Okay, it's a very simple system. The dynamics is underdamped, which means that in this case, you need not only the position to evolve the dynamics, but also need to know the momentum, right? So if I just give you the position, you won't be able to tell me in which direction the particle is going to move, but as soon as I give you the velocity, you can tell me where it's going to go. Right, so this position momentum phase space is Markovian and maximally predictive of the future, right? Given the position momentum now, I can tell you where you're going to go. Now, to emulate a real world sort of data scenario, let's say that we don't know about the physics and we're just measuring the position of this particle, okay? Now, one simple way to get back this additional momentum information and reconstruct the full maximally predictive Markovian state of the system is to just take time delays. And this is an old idea from dynamical systems theory. I'll go back to this in more detail a bit later, but the notion, essentially, you can see it here from a physics point of view, that if I take time delays, I can approximate the momentum, right? And so this is sort of the intuition. And what we're going to do essentially is that we're going to take our measurement time series, expand the state using time delays, and then try to maximize the predictability of this reconstructed state using this transfer operator representation that I was just describing to you in the previous slide. Okay, so essentially what happens as we add time delays is that we, in some sense, narrow down the prediction of the future. So we're using information about the past to narrow down our prediction of the future. And so what you can see here in this schematic is that as I increase the number of time delays, so going here from light gray to dark gray, the distribution of future states given the past states essentially gets narrowed down as I keep on including more and more time delays. And this is naturally captured by the notion of entropy rate, right? Which is basically measures just how spread out is the distribution of future states given the past states. And we're going to leverage this to try to find the number of time delays that we need to get sort of a maximally predictive state. So at some point, you don't need to look further into the past because you're not going to do much better at predicting the future. Now, it turns out that the transfer operator representation I just described to you before, it offers a direct means to the estimation of entropy rates. So in practice, given a candidate state space, what we do is that we partition the state space into small little bins. And then we approximate the action of this transfer operator essentially as a Markov chain. So by estimating the transition probabilities between these microscopic states. Okay. And from that, you can directly estimate the entropy rate. It's very simple calculation, right? So Pij is just an element of this transition matrix. Pi of I is just an element of the steady state distribution that I can estimate from the eigenvector of eigenvalue 1 of this matrix. So this is a very direct estimate of the entropy. And so what we would like to do then is to essentially study how this quantity, the entropy rate, behaves as a function of how finely I discretize the state space, so how many bins I put in there, and how many delays do I take, so this quantity here, k. And so this is what we find when we do this in the double well example. So as I showed you, and it's schematized here on the left, as you add time delays, you essentially narrow down your prediction of the future until eventually, you do as good as you can. And that's captured here by these two figures here. So in the inset, I have the entropy rate as a function of the number of partitions. Okay, and something that I didn't tell you is that in the limit of infinite data, this quantity, the entropy rate, is a non-decreasing function of the number of partitions. So the smaller I make my length scale, so the more partitions I put, the more information I have, because if I have infinite data in a stochastic system, there's going to be always information on all length scales. And so we sort of leverage this intuition to guide our choice of the number of partitions given the limitations of the data. Because what happens is, because I have finite data, at some point I'm going to run into sampling problems. And so what that's going to lead to is an underestimation of the entropy, which you see here very clearly for k equals 1 delay. So this is color coded as a function of delays. And so then we can compare basically the behavior of the entropy rate as a function of the number of time delays. And what you can clearly see is that if I just measure the position, which is this curve here, which is with 1 delay, so basically it's just the position, the dynamics is very unpredictable. But as soon as I start adding time delays, my dynamics becomes far more predictable and the entropy rate drops. And you can see that here very clearly. So n star is basically my choice of the number of partitions, which defines my transfer operator somewhere here. And you can see that as I add time delays, basically I make my dynamics more predictable until eventually, I do as well as I can. So after about 5 frames, I'm not gaining much by looking further into the past. And this is what's going to define now my new state, right? So my state now is a sequence of positions which also captures momentum information. So now that we have this reconstructed state, which we hope sort of captures all the information about the dynamics, we can use this inferred transfer operator that we get directly from doing this estimate to extract the multiple timescales of the system and try to isolate the slow dynamics of the system. And so in this case, it's kind of trivial, right? So it's a particle that is hopping in a double well potential. If the potentials are deep enough, then the longest time scale is the hopping between the two potential wells. And we can estimate those hopping rates directly just by looking at the eigenvalues of this operator that we infer, okay? So here I'm showing you that across a wide range of temperatures. And another neat property of this operation is that the eigenfunction that corresponds to this eigenvalue, which I'm showing you here on the right, it essentially splits the system into the two wells. So you can see that the sign of this eigenfunction identifies the two wells of the system, so the two metastable states of this potential landscape. Okay, so just a quick recap. In a more general sense, our approach is essentially composed of 2 main steps, okay? The first step is to try to use these time delays to obtain a reconstructed state in which the dynamics is Markovian. And so more formally speaking, what we're doing here is the following. So we assume that there is some underlying unobserved dynamical system that we don't really have access to, from which we take some measurements. So this operation here, M, is a function that is going to go from the full state of the system and project it down to the measurement space. And essentially what we're doing is that we're projecting the full dynamics onto the subspace of the measurements and in some sense, sort of marginalizing over these unobserved degrees of freedom. And so inherently, we're going to get some kind of generalized Langevin equation, right? So what we're doing really is formally a Morris-Wanzig projection, okay? In which case, then you're always going to have a inherent systematic source of error that comes from this history dependence, right? Even my best possible Markovian model that only depends on the current state will always have some inherent error coming from the fact that actually there's these memory kernels that I'm not taking into account. Okay, so there, yeah. No, it does vary depending on the underlying properties of the system itself. So there's two things here. So one is the number of bins, and the other one is the number of time delays. And these are two things that sort of depend on each other. And just to clarify, so the number of bins is just basically, how many partitions I put in my state space. The number of delays defines a new space. So, with k equals 2, I have the position now and the position at the past, k equals 3, another point. So it's a three-dimensional space. So the more delays I have, the higher dimensional is the space, and it allows me to disentangle the different dynamical regimes a little bit better. So I think what you were asking is, how do you know how many partitions and how many time delays to take? So following this general property that the entropy rate, if there is higher order dynamical information, it's always going to decay on different rates, but it's always going to drop until basically it becomes a constant, because no matter how further you look into the past, you cannot really make a better prediction of the future. So the qualitative behavior of these curves is kind of independent, especially this one, is independent of the nature of the underlying dynamical process. And so you can just use the fact that you want to basically find where the derivative of this function roughly goes to 0. But it's a really good point. So for example, the behavior of this function here, it's different depending on the underlying dynamics of the system. So for example, if you have deterministic chaos, you're also going to have some entropy production because there's exponential divergence of trajectories. But because now the state space sits on a fractal, there's actually a characteristic length scale below which you cannot gain any more information. So actually these curves become flat at some point. And this is characteristic of chaos. It's just an aside, but so, these things have been worked out quite some time ago, and we can leverage this to sort of understand, how to guide our choice of these different parameters. But there's really basically only two parameters you need to set. So n is set essentially by the finite size effects of the data. So you want to have as many partitions as you can, depending on how much data you have. K, I find, is a bit more of a fundamental quantity, and it depends on the underlying structure of the dynamical system. Yeah. So you're going to need more time delays the more complex or the higher dimensional the dynamical system is. Yes. Yeah.
So, but the thing you experiment is total.
Yes. Exactly.
Control.
Absolutely.
So there is some, right? It might be that you need to go to higher dimensionality.
Yeah, you might need more bins. Yeah. Mm-hmm. Yeah.
The other question is,
why isn't it 2? Yeah, that's because we're simulating this with an Euler scheme, and that has some memory to the actual propagator of the dynamics, because I need to know the initial condition of the position, the momentum, and the noise to be able to define the state, right? Because when I'm simulating with Euler, I'm sampling an initial error. So there's basically at least 3 things that I need to know. So naively would think just 2 delays is enough, but actually it turns out that it's not. Yeah.
So it's some mismatch. It has to do with how the continuum.
Exactly. It has to do with how you discretize the continuum dynamics. Yes. But this is actually, we were at the beginning, we didn't really get this. Why is it not that it's a 2? Until we looked at the precise details of how you actually discretize the Langevin equation. And then you see that actually the propagator has this extra time step.
The other question is, if you have a system that never becomes perfect, so for instance, suppose that the friction of this particle occurred at in principle, this never plateau.
Yeah.
But in practice, what actually happens?
I haven't tried a system like that, but I have seen systems in which this doesn't really plateau. So it decays much strongly initially, but then it's kind of like slowly keeps decaying. And I could argue that this will still allow you to see that at least, you shouldn't be working with your measurements. You should be working with some expanded state. But if you want to model those dynamics, then you need to put in some non-Markovian effect on the modeling of those dynamics. So my underlying assumption here is that there is some finite correlation time such that I can take time delays and recover a Markovian state, which of course has this assumption of a finite correlation time. Yeah, but you will see it in the behavior of this curve though. That's a really interesting question. Okay. So going back here, and so again, as I was saying, this is a kind of a general property of any measurement, I feel, in any kind of field, which is the fact that if you're looking at a dynamical system and you only have time series signals and you don't know the underlying state space, you're not guaranteed to actually be able to find, a neat Markovian description, and you might need to have to deal with this inherent history dependence that comes from the fact that you have a bunch of unobserved degrees of freedom, essentially. Okay, and so there's roughly two ways of doing this, and in the time series analysis literature and also in the deep learning literature, people use, for example, extended autoregressive models or RNNs or something like this, which take into account, memory. What we're going to do here, instead of trying to model this memory explicitly, we're going to change the representation. So instead of trying to build a more complicated model that captures also the memories, we're going to change the representation by including time delays, such that now in this expanded state, my dynamics now becomes Markovian, and I can get away with a simpler description of the system. So that's kind of the intuition, is to really focus on the representation here. And we use also this transfer operator representation that I was describing to offer sort of an information theoretic measure of predictability, which allows us to find the amount of time delays that we need to get this maximally predictive state. And so, the approach has these two steps, the state space reconstruction and working with the transfer operator representation of the dynamics. They're interdependent on each other. And one of the advantages of working with a transfer operator representation of dynamics, is the fact that this representation is invariant to smooth coordinate transformations of your system. So when you reconstruct a state space, and this is kind of old theorems from math, from Takens and Stark and others, you're only guaranteed to recover the local topology of the trajectories, meaning that you can, squish the dynamics, but you cannot cross trajectories. And so, the precise functional form of the dynamics of the reconstructed state is inherently going to depend on how exactly you do the reconstruction, right? So I can make a coordinate transformation here, then that's going to change F. And I want my dynamical representation to not depend so much on a specific functional form or a specific parameterization. And this type of representation allows me to do that a bit more freely. And then, the second advantage, as I said before, is that it's just a linear operator. So I can diagonalize it, I can look at its eigenvalues, and I can extract structure at multiple scales. That's kind of the idea. Okay, so does it make sense to apply this to animal behavior? Well, as we've briefly seen in the beginning, we can sort of think about behavior as, having some stereotype movements that the animal repeats over and over again. So an enticing analogy is to think about behavior as evolving in some complex potential landscape where different wells correspond to different stereotype behaviors or regions of this behavioral space where the animal spends most of its time. And then fluctuations, for example, can drive you to transition from one behavior to the next. Now, in order to make this analogy a bit more precise, what we need to do is to essentially find sort of collective variables that allow me to disentangle between these different states. And this is essentially what the approach that I've just described does. And so we're going to proceed much like as we've done for the underdamped double wall example, but now working from time series of postures of C. elegans locomotion. And so we've known for quite a while now how to go from a video of a behaving worm to a low dimensional representation of the posture. So what that means is that what we do is that we draw a line sort of along the center of the body, and then we can project that into a lower dimensional space. And what that basically means is that by linear combination of these 5 different sort of primitive shapes, I can get back any shape that the animal can do. So I can change these 5 numbers, I get a different shape. And the full space of shapes is essentially captured by these 5 different numbers, okay? So what that means is that I can then transform a video like this into a time series like that, okay? There's something that I didn't mention here, which is the fact that, of course, this representation is invariant, it's translation and rotational invariant. So I don't care where in the arena the animal is doing a specific posture. I also orient all the shapes to be centered around 0. And so this is kind of a translation rotational invariant representation. But so now we have this time series and we're going to treat them as our measurements, as we've done before. And we're going to compute the same quantity as I've done for the double well, which is this entropy rate. And as you can see, basically, if I just look at the posture, then I'm going to have a less, yeah, go, Talmo. Yeah. Yeah. I'm just subtracting the mean, I guess, the mean angle. So I measure the curvature along the body, and then I just sort of align them all to the same direction.
But if the curvature is weird, right? You're not going to get some jiggling.
In what sense? No, I don't think so. I mean, bottom line is basically these 5 numbers. Yeah, the head is always aligned in the same direction. Yeah. And the overall, so the average angle. So if you measure the angle between subsequent body parts and you average that, that averages to 0 across all images, basically. Yeah. Okay, bottom line is that's not enough to capture the state. So if I just give you the shape of the animal, you're not going to be able to tell me what's the future posture of the animal, right? So I need to have at least some sequence of postures to be able to know, for example, whether the worm is going forward or backwards. If I just have an image, I don't know. And this is what this entropy rate captures essentially, is that you need to have at least some time delays before you find the maximally predictive state. So you can see that, you know, at least within error bars, these curves essentially collapse, meaning that using roughly 10 frames, so about half a second of posture sequence, I have reconstructed the state. So now my state space is going to be basically, you know, this 11 frames times 5 eigenworms, so these different primitive shapes. So it's a 55-dimensional space. And this characterizes the behavior of the worm. And so one thing that I can do just to visualize the space is to project it down into 2 dimensions. So what I'm doing here is just leveraging, yeah, go ahead. Sorry? Yeah, so here, this is the sampling problem that I was telling you about. So because we don't have infinite data, the entropy rate starts decaying at some point because I'm not managing to sample that. So this also helps us set the number of partitions, which is roughly going to be around here, around 1,000 partitions of a state space. So we're basically transforming this continuous dynamics of the worm into a Markov chain with 1,000 states effectively. This is what we're going to do. Okay, but let's stay within the continuum yet. Yeah. Yeah. But the space is not really 55-dimensional. I'm embedding it into 50. Huh? Yeah, so I do a Voronoi tessellation of the space, actually. I didn't speak in detail about how you actually do it, but so there's 2 steps. You do a Voronoi tessellation while maximizing the entropy rate, actually. So you reinitialize so you basically sample random points in the space, and then you basically make a Voronoi cell. And you do this multiple times until you find the maximal possible entropy with respect to the partitioning scheme. So we are making a particular choice of partitioning scheme, and you might wonder what are the implications of that. In the case of the C. elegans locomotion, as I'll show you in a minute, this model does pretty well at capturing the behavior, but that's something to consider in other systems. Yeah. Cool. So it's a good point you're making. The state space is not really 55-dimensional. I'm embedding it in a 55-dimensional space. Actually, previous work that we've done has shown that actually within, order 10 dimensions is the real dimensionality of the behavioral state space. We're just embedding it in a much higher dimensional space, which allow us to disentangle behaviors much better, actually. So by blowing up the dimensionality, you can see the different behaviors in a much neater way. So what I'm doing here is just learning sort of the local geometry of the space and projecting it down into 2D using a UMAP embedding. And you can kind of already see that there is some structure to this space. And this structure becomes even more clear if you start color coding this by some kinematic properties like the velocity of the animal or its body curvature. And so on the left-hand side, I'm color coding it by the velocity, where you can see that there are these sort of quasi-periodic orbits here that have a positive velocity. So this is when the animal is moving forward. In this region here, for example, we have also nearly circular orbits where the animal is moving backwards with negative velocity. And then you kind of have these external lobes here on both sides, where if you look at the curvature, you see that one of them corresponds to the worm bending towards the ventral side, so when they bend towards their belly, and the other one corresponds to the worm bending backwards, so towards the dorsal side. And so you can already see that, different regions of this state space neatly correspond to different types of behaviors, although these are very short timescale behaviors. And we were interested in looking at sort of more longer timescale structures. And so we take this data of these worms, which are foraging in a sort of empty environment, and we look at now the eigendecomposition of the inferred transfer operator of the dynamics. And what we find is that there are timescales that relax on timescales of order 10 seconds, which it's much longer than the body waves or these short timescale behaviors, which are roughly half a second. And so it turns out that these long timescales correspond to different states that the animal actually uses for foraging or for navigation. So what I'm showing you here is the same space as before, but now color-coded by this slow mode of the dynamics, okay? Where you can see that when this eigenfunction is negative, essentially the animal is moving forwards, and when it's positive, it's basically doing everything else, which means combinations of reversals and turns. And so these are now sequences of short-time behaviors, so they're much longer lived than before. And you can also see that structure very neatly if you look at the actual trajectory of the animal on the plate. So here it's the centroid position of the animal color-coded by the same eigenfunction, where you can see that essentially you have this sort of nearly straight paths interrupted by these reorientation events. So it reminds you of a run and tumble like description, okay? Except that here it's for a multicellular organism with a brain, and the tumbles are actually collections of sequences of reversals and turns, okay? Also, on top of that, identifying these states, I didn't use any information about the actual position of the animals on the plate. So I'm working directly from this animal-centric posture point of view, which takes out translations and rotations, and yet you get back this sort of larger scale behavioral states. And so if we, for example, partition along this eigenfunction, then we find roughly these two sort of metastable states, and the timescales at which the animal stays in each of these states are roughly captured by two sort of exponential timescales. A longer one that extends to hundreds of seconds, which corresponds to these run states, and the shorter one on the order of 5 seconds that corresponds to these collections of reversals and turns that the biologists have called pirouettes. Okay, so we see that this allows us to do this coarse graining of the dynamics through the spectrum of the transfer operator, but the operator itself is also a model of the dynamics that we can use to sample now, right? And so what I'm going to do now is that I'm going to basically diffuse in this high dimensional state space. So I sample from a specific state, I hop to a different state according to the transition probabilities, and then I sample a new state inside that state. So basically I'm diffusing while obeying this transfer operator dynamics, okay? And so this allows me to make simulations of the posture dynamics over time. And I'm going to show you some results of what that looks like. So these two videos, one of them is a simulation and the other one is data. So what I'm showing you is the body curvature of the animal as a function of time. So basically the angles along the body is a function of time. And one of them is the data simulation. And to be honest, to me, it's a bit difficult to say which one is which. It turns out that the top one is the simulations and the bottom one is the data. But the bottom line is that we capture sort of the statistical properties of the dynamics quite well. So if you measure, for example, the temporal correlation function of the projection onto these eigenworm time series, so our measurements that we started with, you can see that we can capture the correlation function of the dynamics quite well, statistically speaking, through our simulations. On top of that, we can look at sort of longer timescale properties of the dynamics. So for example, I can try to estimate if the animal is one of the states, how much time does it take before it transitions to a different state? So I can compute these hopping rates between these macroscopic sort of metastable states. And I'm showing you here the results of simulations versus data across multiple organisms and these two states, where you can see that we do a pretty good job at predicting the hopping rate between runs and pirouettes. And on top of that, also, what we can do is that we can take into account the hydrodynamic interactions between the body and the agar surface where the animal is, so that then we can transform this posture dynamics into actual propulsion in the laboratory space. So we can make these worms propel. So we start from our simulations of posture and we get back sort of the position of the animal as a function of time. And so we can now generate the random walks that we've looked at in the beginning of the talk, right? So if you remember, the first video we had was sort of the Petri dish on the right-hand side and the microscope on the left-hand side. This is kind of the same thing, but completely in silico, and it's just a full simulation. Okay. And so these random walks already kind of look reasonably similar to what I was showing you before, but we can also, just quantitatively analyze whether we capture the statistical properties of these random walks. And what we've known for a long time is that these animals, they exhibit sort of nearly ballistic motion on short time scales and diffusive motion on long time scales. So if I estimate the mean square displacement, so basically on average, how far do I go in a finite amount of time? There are these two regimes, and you can see that essentially we capture the full ballistic to diffusive transition over 3 orders of magnitude in time. And on top of that, we can estimate, for example, the diffusion constant. So if you go into the diffusive regime, you can just estimate the effective diffusion constant. And it turns out that we can predict the diffusion constant of these animals using this very simple model. And so this is quite a remarkable jump in scale. So we start from a microscopic dynamics at the level of posture, right? And we go all the way to the diffusion in a behaving animal. And this is done using a very simple model, actually. It's really just a Markov model. And this is only possible, I feel, because we've thought very principally about the representation. So we spent some time thinking about how we should represent our system such that we can simplify our theory. And it turns out that if you do that properly, then you can go and do these types of predictions very neatly. That's a good point. So if you just use the posture and don't do the reconstruction, then the simulations are going to be horrible. If you just build a Markov model on that, it doesn't really predict anything. So something that I didn't put here that I could have, it's something that you can do also is that you can keep the state space, but instead of involving the dynamics according to this Markov chain, you can just sample from the steady state distribution. So you do some kind of Gibbs sampling of the steady state distribution. So at each state you sample, some random state, and then also you're not going to be able to predict any of this. Yeah, there's a lot of perturbations you can do here also. So for example, this Markov chain is irreversible. And so what you can do, for example, is to reversibilize the dynamics, make the system more equilibrium-like, and then look at the results. And also there are some qualitative shifts that you find once you reversibilize the dynamics. So for example, the timescales of transitions become much longer, which makes sense because you're forcing the system to be in equilibrium. And so a lot of the properties change slightly. So you need to have the precise state and the precise dynamics in order to be able to recapitulate these results. Yes.
So you showed us examples of transition rates between different parts of the data. But in the lead up, you said, well, you also have this nice structure that you would expect from like. Sometimes that's not how you did it. It seems like you did it by actually drawing a box.
Yes. So
you didn't show us the spectrum. See, this rate is good. You showed us in this eigenfunction, this region is this behavior, this region is this behavior. But you didn't show us the spectrum and say, this
yeah.
is
Let me see if I have that figure actually somewhere. So that's an estimate that we have, and this eigenvalue is going to correspond to sort of the inverse of the sum of the rates of transitions. I don't have the estimate here, but I can show you from the paper, and they match. So you can get the hopping rates from the eigenvalues, except that the eigenvalue doesn't give you the hopping rates. It just gives you sort of the sum of the inverse of the sum of the hopping rates. So it's related to it, but doesn't directly give you the hopping rates because this system is very asymmetric. Right? So if you have symmetric wells, then it's fine. So it's just the half of that. So that's also why I could do it for the double well, because the well is symmetric. And as you can see here, there's an eigenvalue. So yeah, so there's an eigenvalues, and there's a quite large timescale separation between the first eigenvalue and the second one. And then from then on, they're roughly equivalent to each other. So at least in this setting with this data, this is what we find. If you look at different experimental conditions, for example, if you put the animals on food or you give them stimuli or something like this, you're going to modify the full spectrum. And this is kind of a phenotype that you can leverage.
So there really is
yeah. Yes. In this specific case, yes. Yeah. There are cases where it's not as clear. Yeah. But you can always, my intuition is that you can always isolate the slowest time scale and effectively treat, so you can do some kind of time scale separation. Let's say now I'm just going to sample my dynamics on this sampling time and treat basically all the faster dynamics effectively as fluctuations. That's kind of the intuition behind what we're doing here. So I spent a lot of time doing this because actually what I wanted in the first place was to be able to isolate some kind of collective variable that I can treat as some kind of like order parameter like thing that I can now build a model of, because starting from videos of animals is quite difficult to build a principled model. But if you can isolate variables that are sort of capturing larger scale properties, it becomes easier to now start thinking about the right model class for these types of variables. And I'm going to show some of that in a minute. Are there any other questions here? Okay, if not, then I'm going to continue. So as you're probably, itching to know, this simple Markovian description of behavior cannot be complete, right? It's impossible that this is the full story. We know that the animals have much longer time scales, there's aging, for example, and all sorts of things that we're not capturing with this description. So this description does quite well, but it's not the full story. And there's actually a couple of subtle statistical properties that we do not capture with this description, and I'm going to go through those in a second. So one of the things that we cannot really capture, I'm going to show that to you later, is the fact that this distribution of first passage times exhibits a slightly heavier tail than what we can predict with our Markov model. And another property that it's much more non-trivial, I find, is the fact that if I project all of my dynamics onto this slow mode now, and I do this time scale separation, and I only focus on the dynamics of this slow mode and compute the autocorrelation function of this slow mode of dynamics, what I find is that these correlations decay at least initially as a power law. And this cannot be attained by a finite Markov model, which will have a correlation function, which is sum of exponentials. Right? So there is a bit of an enigma here. On top of that, actually, if you look further in time, of course, because this is a finite data set, the correlation function eventually crosses zero. Right? So there's some finite correlation time that will be defined by the zero crossing. And so we can probe this kind of apparent scale-free like nature of the dynamics by studying the finite size scaling of the correlation function. So what we're going to do is that we're going to do something like a Kadanoff block spin transformation where we average the dynamics locally and we recompute the correlation function, which is going to give us a new correlation time. But if you rescale all of these correlation functions by their respective correlation times, these correlation functions collapse. And this, seems to indicate some sort of, scale-free dynamics or criticality. Yeah. That's why I wanted the blackboard. So we have this high-dimensional state space. I compute the eigenfunctions of this transfer operator. And then what I do is that I basically project all of my dynamics onto those eigenfunctions. So then at each time point, I can project the state onto this eigenfunction. So it becomes now sort of a one-dimensional time series of just the projection along this eigenfunction. It's going to be basically staying within the run state, then hopping to the pirouette state, then going back to the run state. So I'm projecting this dynamics onto this kind of effective manipulation by the variable. Okay. And then I'm looking at the properties of the dynamics of this slow mode. And when I estimate the correlation function of that, I find that there are these very long range correlations, which I cannot see when I just look at the eigenvalue correlation before, that they have this oscillatory pattern. There's no interesting dynamics there. But if I look only at the slowest possible ergodic dynamics of my system, which I can extract from this transfer operator representation, then I find these non-trivial correlation structure, these long-range correlations in time. Yeah, so here what I'm doing is that I'm averaging the dynamics locally in time. So I'm taking sort of increasing time window lengths, sort of downsampling my system in time, and then looking at doing a local averaging of the dynamics there, and then recomputing the correlation function. But now, you know, it has been downsampled in time, so it's going to give me a different correlation time. And then what I do, yeah. No, so this is a temporal cross-graining of the dynamics.
But the thing that you're averaging
is the projection onto the slow mode. So,
but then in that case, okay, alpha 2W is a linearly fixed versus alpha.
in a way. Yes, so and basically, yeah. And I'm also doing a rescaling operation here by the correlation, the new correlation time that I find. Right? So there is an averaging and a rescaling. Yeah. Okay, so yes. Why are they becoming increasingly anti-correlated? Yeah, I'll get back to that in a minute. Yeah, so this is kind of weird, right? So you have these long-range correlations. This is the full experiment, actually. So 30 minutes is basically the length of the experiment. And you consistently find that at very long range, you have these anti-correlations. So I'm going to get back to that in a minute, but this is a really good observation, actually. So we find these non-trivial correlations that we cannot explain with our model. I'll show you that in a second. And so then I started thinking a bit more, you know, from an abstract point of view, you know, how can you generate dynamics of this sort? And so, you know, we know that in behavior, there are slowly varying internal states that are modulating the behavior on very long timescales, right? So for example, neuromodulation can change sort of the effective interaction between neurons on very long timescales. So it's almost as if like the entire model is changing over time. And so what we're going to do here is that we're going to take this notion very seriously, and we're going to see what happens when we do have such sort of slow, non-ergodic drives that prevent the system from ever relaxing to a steady state. And so in our kind of a potential landscape picture of behavior, you can imagine that the potential landscape itself, it's being slowly shifted over time on timescales comparable to the observation time of the system, okay? And so generally speaking, what we're going to be looking at here is this type of model class where you can think of X as my slow-mo dynamic, so this emergent sort of collective variable that evolves on some potential landscape, and the potential landscape itself is being driven by some parameter alpha, which I assume to be slow. So I'm going to set this time scale to be really slow, okay? And then we're going to look at what happens when you have dynamics of this kind. And one of the things that we find, and I'm not going to go in detail onto that, I'm just going to show you the result, is the fact that regardless of how exactly you define the potential and the fluctuations of the parameter, as long as the potential fluctuates slowly enough, you will always find heavy-tailed first passage time distribution. So if I compute the amount of time a particle stays in the bottom of a well, or the amount of time it takes to hop out of a well, it's going to have this type of structure where actually if I increase the temperature of the fluctuation, so if I make my potential fluctuate slowly, but also a lot, in the limit of large temperatures, then I'm always going to get a power law with an exponent of -2, exactly. And this has actually been found in many systems, in behavior and beyond. And we here show that this can be explained by this type of picture where you have this sort of non-ergodic drive that forces the entire system slowly and with large enough fluctuations. So let me just give you a very simple example of dynamics like this. So let's say that you have an overdamped harmonic oscillator. So you have just a quadratic potential and the dynamics is relaxing to the bottom of this potential, and that the potential itself is being shifted by this variable S. So I'm moving the potential around back and forth. So while the particle is relaxing to the bottom of the potential itself is shifting around, and it moves closer or further away from an absorbing boundary that I put here. So when the particle reaches that boundary, it basically, disappears into infinity. And so in a system like this, if we estimate the first passage time distribution, we find that, so for low temperatures, what I'm showing you here, so this is the distribution function of the first passage time. So from red to blue, you have what you would find if you just have a fixed potential that it's not changing in time, in which case you just have an exponential function for the first passage time distribution. And red is when you're closer to the boundary, so you reach it faster. Blue is when you're further away, so you reach it slower. Okay. And then here color-coded from like dark blue to yellow, you have the result of what happens when you allow now the potential to be fluctuating in time. In two very distinct regimes, one in which the potential is fluctuating very fast, and one in which the potential is fluctuating very slow. And both of these dashed lines are theoretical predictions that we can derive analytically. And what you can see is that if the potential is fluctuating really fast, then you essentially get back the average behavior, because essentially the potential is going to be at the mean value most of the time, by the time that a particle actually reaches the absorbing boundary. Whereas when the timescales of the fluctuations of the potential start becoming slower and slower, then you start observing the emergence of a heavy tail. Okay. And in the limit of large fluctuations, then you actually see this exponent of -2 that I was telling you about. So when I increase the temperature, then I always get this weird sort of power law-like first passage time distribution. And so it seems like we might be able to at least explain these heavy tails we saw in behavior if we allow for this fluctuating potential landscape picture. But remember, there was also this weird behavior of the correlation function, right? And in fact, if you just have a system as simple as this, where you just have a harmonic oscillator that is being driven, the correlation function is going to be very simple. It's just going to decay as a sum of two exponentials, right? A fast one just corresponds to the behavior of the particle, and a slow one that corresponds to the moving of the potential. Okay, so it doesn't seem like we can capture that weird behavior of long-range correlations and longer-range anti-correlations. Where is all that coming from? So the issue is that when we estimate correlation functions, we do it through time averages, right? And this quantity here is estimated through ensemble averages. And so we need to take into account the fact that there might be some non-ergodic finite size corrections to the correlation function that we're not taking into account. It actually turns out that after re-deriving this result, I found out that Jonathan Depont back in 2016 had already derived an expression for the finite size correction to the correlation function, which looks like this very nasty beast here, but where you can kind of see that, so L here is the time lag of the correlation function, where you can kind of see that because of this negative part here, there is a regime where you can have the correlation function actually going below 0. And this is what I'm going to show you next, which is the result of numerical simulations versus the prediction of the theory, based on this finite size correction to the correlation function, where you find three distinct regimes that I find are kind of interesting. So what I'm showing you here is color-coded is the correlation function. So red is when it's positive and blue is when it's negative. The x-axis is the sort of the transition time, or the lag time of the correlation function divided by sort of the experimental timescales of our system. So how long am I observing the system? And on the y-axis, you have the ratio between the fluctuation of the potential and the time scale of the observations, okay? And what we find, what? Oh, sorry. Yeah, okay. Yeah, anyway. Cool, so what we find are three distinct regimes. So one of them, you basically recover the ergodic limit. So when the time scales of the fluctuations are much shorter than the observation time, then you basically get back sort of the result that you have an exponential decay to 0, roughly. Whereas when the timescales of the fluctuating potential are super long compared to the observation time, then within the observation time, basically the potential is frozen, right? So it's so slow that you actually don't see this slow time scale, in which case then it just relaxes to 0 on the time scale of the fast model. But then if the timescales are comparable, if the potential is fluctuating on timescales comparable to the observation time, then you start getting this non-trivial behavior here, where, as you were pointing out very correctly, you have this long-range anti-correlations emerging. And so it seems like having a non-trivial interplay between the timescales of observation of the system and the intrinsic timescales of the dynamics can give rise to this non-trivial behavior of the correlation functions. Now, does this actually explain what we saw in the worm behavior? So let's go back to the worm data. And so what I'm doing here is that I'm projecting the dynamics onto the slow mode, and I'm trying to learn some kind of, Langevin equation for this slow mode through. So I do a timescale separation such that the noise now I can treat it effectively as white, and I can try to fit this drift and diffusion model to the data. Okay. And what I'm going to do here is that I'm going to allow the potential to change in time. So I'm going to do this inference sort of in time windows. And so as you can see here in this plot, so this is the potential as a function of time. And I'm just reminding you what these different regions correspond to. What you see is that as time goes on, that the animal actually is biasing the behavior towards the run state. And this kind of makes a lot of sense because these animals, they're put in an environment where there's no food. So they're starving. Oh, it's flickering again? Oh, it's going. So they're starving, so they're biasing their behavior towards the run state because they need to go further away from where they are right now because they realize that there's no food where they are. And so there's this very slow drift biasing the dynamics towards runs, which is probably what you guys are going to do soon because it's reaching lunchtime and there's no food here. And so, to assess whether this might account actually for these non-trivial observations that we see, we're going to make some simulations, okay? And what we find is the following. So if I just take a stationary potential description, so sort of an ergodic description of the system with a fixed potential landscape, then what I observe is that I actually do a pretty good job at capturing the timescales of transition in between the states, except for a few points in the tail of this distribution. And I also do a good job at predicting the correlation function, but only for very short timescales, like within a minute or so. And then I relax to 0, and whereas the data has this sort of non-trivial behavior. Now, if I allow my potential to change in time, so if I do this inference where now the potential is also explicitly a time-dependent quantity, then I start capturing both this heavy tail in the distribution, but also this non-trivial skill-free-like behavior to the correlation function. And so it seems like, having such a potential landscape that fluctuates on timescales comparable to the observation time of our system can give rise to these non-trivial statistical properties of the dynamics that we see in the behavior of C elements. And we think that this is actually probably a general property of behavior across scales. And it might also explain a number of observations on non-trivial behavior across organisms and even across biological systems where multiple time scales are ubiquitous, right? So I think, yeah. If I have a physical interpretation of this potential. So the potential essentially, you can invert it and get a sense for the probability of doing different types of behaviors and looking at sort of the time scales of transitioning. So the deeper this well is, the more time the animal is going to spend in the run state. The deeper that well is, the more time it's going to stay in the pirouette. You can see that it's actually, a rugged potential. It's not as simple as a double well. But that's sort of the interpretation. Absolutely. The shape of the landscape defines the dwell times and different behaviors. Yeah. And as this shows, if you do that properly, you can capture actually the dynamics pretty well. Cool. Are there any questions on this? I understand it's heavy and there's a lot of stuff. Huh? The rescaling of with the model, you mean? I haven't tried, but I'm guessing yes. I think I've done it. Yeah, but that's a good point. I should look into that. Yeah. Cool. I think I'm gonna just leave you at that actually, unless you want me to discuss some other stuff, but I think this is kind of a nice bulk that we can keep discussing over the next, the rest of the day. This is basically most of what I wanted to say. I hope I motivated you guys to come thinking about behavior from this physics point of view. There are a ton of interesting questions and more high quality data like Talmo has showed you. And I'd just like to end by thanking Bill for the invitation. Thanking you guys for your attention. I'll be happy to take any questions now. Thanks. Yeah. Mm-hmm. So the intuition here is that actually the bulk of the process is in the time delay embedding. So the hope is that, given a general enough observation of your system, that through the time delay, you might be able to get back the underlying state. So even in a higher dimensional system, if you take different functions of the state and drop them down into lower dimensional space, you can always reconstruct it back as long as these variables are coupled to the other variables. So taking 3 eigenworms, 4, 5, 6, 7, it doesn't really matter. As long as you have the right number of time delays that allows you to find the maximally predictive state, then you're good to go. That's a good question. More questions? Mm-hmm.
Then of course, in any particular experiment, it would look like a little bit.
Yeah.
Like how do you think about, when do you think about wiring time code? When you think about so you said that in this case, think about it as shifting from one thing to the other, and there's a natural corollary.
Yeah, no, that's a very good question. And this is kind of it's a deep one too, in the sense that in principle, through the reconstruction process that I'm doing, so this time delay embedding, if I have a general enough observable of my system, then this process should give me the best possible ergodic dynamics. So by ergodic, I mean that I'm going to visit all of the state space and I have some invariant steady state distribution. It's not going to capture anything that evolves on timescales longer than that. And so this is going to give me the best possible ergodic description. And now, when you're working from a model-based point of view, of course, actually in the system that I was working on now with this harmonic oscillator that I'm driving, I know that the system is actually has these 2 time scales, right? But just because I cannot observe the long time scale properly, because there's this weird thing where the time scale of the changing of this variable is comparable to the observation time of my system, I cannot disentangle the two. And so there I really need to have an explicit time dependence to my dynamics. So I cannot get away with just an ergodic description. Of course, it's always going to be a question that depends on the specific details of the system that you're looking at. If you do have a model, then these questions don't even arise. If you're trying to learn dynamics from data, then these questions start becoming important, and you need to take those into account. Yeah. Yeah, that's a good point. So totally. Actually, we've looked at much longer timescale experiments in C. elegans. So we have these like 10-hour long recordings of the animal and food. where the dynamics that you get are actually not this run and tumble like thing. You get much longer timescale structures which correspond to the worms either looking for food actively or not looking for food actively. And these are things that evolve over timescales of decades of minutes instead of this. And these dynamics are actually underlying. So you can think about this in a hierarchical structure where the longer timescale dynamics of looking for food versus not is kind of like at the larger scale. And then within the looking for food states, you find these dynamics sitting there. And these now become stationary. But also in that experiment, we still find a slower time scale. I feel like no matter how long I'm going to record an animal, there's always going to be a longer time scale. So I still find the same type of structure in the correlation functions, the same types of scalings and everything. Actually, it's even more clear in that data. So I guess unless you measure from birth to death, it's going to be difficult to disentangle all of the time scales, really. Yeah, it's a good question. Yes.
And you described this evidence for the being discrete modes of behavior versus
so there is some discreteness to it in the sense that we're calling these states roaming and dwelling states. So the dwelling state is when the animal sort of sits within like close to a food border and is eating for a little bit. And the roaming state is when it's like, okay, I'm fed, I'm gonna look for different regimes. And you can see that these two things, they occupy different parts of as a state space, which is going to be different from this state space, by the way. So there is some sense of discreteness, and you can always also treat now these new longer timescale macroscopic states in the same way that I've treated this run and tumble like states here. So there is some sense of discreteness, which you get from the spectrum of timescales, right? So if there is a timescale separation that tells you that these states are in some sense discrete, meaning that the animal is lingering within these states for a longer time, then it transitions out of them. So that's the sense in which you can identify discrete states on top of an underlying continuous structure. So that's the thing here is that we try not to draw cuts all the way from the beginning. We try to keep as much information about the continuum underlying dynamics and try to extract the structure sort of as a post hoc type of analysis while keeping the microscopic dynamics still there. That's the only way in which you can get a model of the dynamics that captures both the fine scale posture movements and also the larger scale structures. Yeah.
But I know it's unfair to ask you about before you did this, you think of trying to reconstruct the anomaly that had. And you found it. So I was very thinking about it. Maybe you want to open the book.
You want me to tell you this story? Okay, I can. Yeah. 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 Yeah, okay, sure. So basically, I mean, the underlying idea is the same. So we do the same kind of thing where we start from postures, and we do the same thing where we take time delays to reconstruct the state, except that here we weren't using this more information theoretic derived quantity of the entropy rate to measure the predictability. We were defining this quantity that we call prediction time, which essentially measures how much time sort of two neighboring points diverge in the state space. And it turns out that if you actually follow two neighboring points over time, you find that these trajectories, they diverge exponentially in time, at least on short time scales, which is indicative of deterministic chaos. Okay, so, and then you can measure the rate of expansion, and that gives you the Lyapunov exponent. And what we found by doing this type of calculation is that we derived the number of time delays that we need, and also the minimal sort of dimension to the state space. This is what I was telling you before, where you see that roughly, after about eight dimensions, I'm not doing much better. So I can embed this 55-dimensional space that I was looking at before. Actually, I can embed it into an 8-dimensional space without losing any predictability. So this is sort of the minimal set of states of the animal. And this is what we're kind of illustrating here. So these are sort of spatio-temporal states in a way, right? So here you have arc length of the body, so from the head to the tail, and in time here, sort of from 0 to k star, which is the number of frames that defines the state, where you can see that these different modes actually already correspond to different behaviors. So this one, you have a wave propagating from the head to tail, so that's moving forward. This one, you have a wave going from tail to head, so it's moving backwards. And these ones correspond to the worms turning. And so if I now project the dynamics, instead of doing the U map, I can project it into this space. You see that these trajectories have some neat structure, and I can characterize that structure by looking at the Lyapunov exponent. So the idea is that I'm going to look at local neighborhoods of points and seeing how these volumes are sort of sheared by the flow, modified by the flow. So the general intuition is that I start with an initial ball of points, and then as the dynamics flows, as time goes on, this is going to distort. And what I can do then is that I can estimate the rate of expansion and contraction of these volumes. And this is given by the Lyapunov exponents. And I'm going to have as many exponents as dimensions that I have in my system. So let's see, I think here I'm using a 7-dimensional state space. And what we find when we compute these quantities, which is kind of striking, is that, okay, so first, before I jump into that, I should say that the behavior of the spectrum of the Lyapunov exponents is telling you about the class of dynamics that you have in your system. So for example, if you have a positive exponent, you have chaos. If all of the exponents are negative, you have dissipation. You're going down to 0. And you can actually measure the rate of dissipation by looking at the overall sort of rate, this difference between rate of expansion and contraction in the phase space. And for example, if you have a full energy conserving system, a Hamiltonian system, then you need to have the same amount of expansion and contraction of your state space because you need the overall phase space volume to be conserved. That's just Little's theorem. And so when we measure this, we were intrigued by the fact that the spectrum is symmetric. So it has signatures of something that looks symplectic or equilibrium-like, except that the axis of symmetry is not at 0, as you would get from a Hamiltonian system, but it's actually shifted towards the negative side. So it's almost as if the system is like Hamiltonian, but not quite. There's an overall rate of dissipation. And this is indicative of a specific class of dynamics, which is that of damp-driven Hamiltonian systems. And this is something that it's still enigmatic to us, and we would like to, try to write down a model or, a biomechanical model that has this type of structure. And I'm not sure, to answer your question, Bill, I'm not sure exactly how, this structure relates to what I found in terms of this transfer operator representation. These are kind of equivalent ways of looking at the dynamics. This one is more French scale trajectory based. The other one is more sort of larger scale ensemble based or probability based. They're equivalent to each other, but they highlight different properties of the dynamics. When this one highlights more sort of finer scale properties of the dynamics, the transfer operator representation captures more sort of larger scale properties of the dynamics. And it's not so trivial to do this mapping. I mean, even in known dynamical systems, it's very non-trivial to do this, especially when you're in presence of a chaotic attractor like this with damp-driven Hamiltonian structures. So if anyone wants to think about this, I'd be very happy to discuss. But this is it's some intriguing structure that we find when looking at the behavior of salients. Yeah. Cool. Any questions on this? If they depend on how you find the dimensions. No. Although, I mean, so these results are specific to this specific data set where the animals are foraging for half an hour. We haven't looked in terms of this type of analysis on other data sets. So I'm guessing the structure of the state space is going to change. So for example, if you have a mutant, something that I didn't tell you about is that you can actually disrupt this crazy behavior of the correlation time on the first passes time distribution by knocking out a neuropeptide receptor. So there's this specific class of neuropeptide receptors called NPR1. And if you look at worms that lack this receptor, actually they exhibit the same behavior on short time scales. They still do the same type of forward, backwards, and turns, but they don't exhibit this much longer time scale behavior somehow. So we can use all of these things also as phenotypes to try to understand behavior. And in this specific case, I don't have a figure here, but we can actually knock out this long time scale by genetic perturbation. So this sort of thing we have done. Yeah. And it would be interesting to see how, for example, this whole spectrum changes as you perturb, the animal in different ways. Yeah. Cool.
Awesome. All right, thank you guys. Yeah, so this is ostensibly a workshop on the physics of behavior. My training is engineering. I worked in my PhD in a center that had a good number of physicists working on neuroscience. What I'm going to be talking today about is mostly dyadic behavior, interactions of pairs of animals. And physics for a pair of interacting animals is kind of a questionable thing. I think Gordon Berman has given some nice talks on this. If you're thinking of like big groups of animals, treating them as a swarm, that's something you can kind of model. If you're looking at the behavior of a single animal, that's something you can kind of model. When you have pairs of interacting animals, a lot of the details matter, and getting to something that you could call physics, I'm not sure we're quite there yet, but we can maybe do a little bit of math of describing what animals are doing. So there will be a little bit of math in these slides. And then I also wanted to just talk about some of the neuroscience of social behavior and some of the things that I've worked on recently and directions that I think are interesting in this space. So I mostly come from neuroscience, and I'm interested in this question of understanding what's happening as we're interacting with the world around us, what's happening in our brains. And the way that I always saw this phrased when I was in grad school was that you have some sensory input, you process that sensory input, you perform some sort of action selection on that, and you produce a behavioral output. And there was this, depending on the type of neuroscientist that you were, you might focus in on the sensory processing thing or the motor control action execution thing. And then there was this thing that happened in the middle that we didn't really think very much about. And for a long time, I think neuroscience was largely driven by this attempt to characterize these two outer parts of the brain that are doing the processing and the motor output. And if you look back to, say, the 80s, we've kind of established this school of reductionist neuroscientists, starting from, say, Hubel and Wiesel and Sperry, where you'll take an animal, you'll record from some part of the brain, maybe that animal is awake, maybe it's not, and then you'll present a battery of different stimuli to the animal, and you'll see how that neuron responds, and you'll produce a receptive field. And this theory of approaching neuroscience still kind of exists today. It's become a little bit more technically advanced. We can look in more brain areas at once, but we're still kind of recording and correlating and trying to make sense of how the actions of the animal relate to what we're seeing in the neural activity, and kind of reduce it down to this relationship of these two signals. And we've gotten some theories that have come out of that. We have these ideas of deep and convolutional neural networks that were inspired by the visual system. We have theories of how you encode information, building sparse representations of signals in some brain areas, doing predictive coding or optimal coding of signals. And we have some theories of how you decode information from a big population of cells that's representing some combination of sensory cues in the world. This has mostly been done in the framework of still a head-fixed animal, maybe walking on a treadmill and being passively presented with stimuli or being presented with very simplified stimuli. And this started to change maybe around a decade ago when we noticed that, or I guess when there were two kind of factors that changed our perspective away from this kind of Hubel and Wiesel just batter neurons with a bunch of stimuli and see how they respond and build a model out of it. And it was kind of a push-pull system. So on the push side, we started to realize that if you record from neurons, even in, say, primary visual cortex, and the animal changes what it's doing, it goes from being stationary to running, the responses of those neurons change. So even though this is ostensibly a sensory processing area, you see this modulation of the brain's activity depending on the state of the animal. So there was this clear evidence that you couldn't just record everything in an anesthetized animal and then from that distill out an understanding of how the brain works. But the behavior that an animal's doing influences computation across a wide variety of brain regions, including ones that you wouldn't think of as generating behavior. And there was also a pull because we began to kind of miniaturize our recording setups, and it became easier to study the brains of animals as they're running around and interacting with the world with head-mounted microendoscopes. And we could generate transgenic animals to record from different genetically defined cell populations and manipulate their activity and look at an effect on behavior. And on top of that, as you guys might have heard, there have been some software tools developed for automatically detecting the posture of behaving animals and quantifying that posture and extracting out features of how animals are moving and what they're doing. And these provide an additional rich representation of how animals are moving that we didn't have access to before, that kind of makes the problem of studying running around freely behaving animals a little bit more feasible. So this has driven a kind of change in the way that neuroscientists have approached behavior. And it's pushed us away from this kind of very rigid, simplified setup where an animal is maybe trained to press a lever in response to a light stimulus. And it's pushing neuroscientists more towards, or at least some neuroscientists, more towards a perspective of understanding the kinds of behaviors that brains evolved to produce. Social behaviors, interactions between predator-prey species, raising of your young in some cases, and the structures in the brain that give rise to these complex behaviors. And there've been, at this point now, a good number of review articles saying that we need to start thinking about these things. We need to start thinking about not just the behaviors that we can train animals to do in an operant box in an input-output way, and these like reductionist motor control tasks that we can do, but that we should also think about the kinds of things that brains have been genetically wired to do and the things that they need to do in order for an animal to survive and reproduce. And you can still be reductionist about this, but it's different from having a drifting grading and changing its contrast and its orientation and seeing how a neuron changes its response. You kind of have to just look at what animals are doing, try to quantify that and try to relate it to what the brain is doing. And the things that animals do on their own without any training can be pretty wild. So this is an Argus pheasant showing a courtship display towards a female here. And there's an interesting combinations of things going on. There is a visual processing pathway that's taking place here. He's identified that this is another pheasant. There's a motor control aspect to it. There's also probably a learning aspect to it. He's probably done this a couple times before, and whether he's willing to do this or not is going to depend on his level of maturity, if he's juvenile or adult, and also how healthy he's feeling, if she's being receptive or not. So there's this whole area of understanding this kind of behavioral decision-making that has emerged in recent years in neuroscience and gotten this renewed level of attention. And it's a very different flavor from kind of the corticocentric neuroscience that I thought about a lot in grad school. And it leads to some really interesting questions about how you design a system to produce these behaviors in a way that is flexible and adaptive, in a way that you produce the behavior at the appropriate time and that you execute that behavior and that it leads to some beneficial outcome to the animal. So people have thought about these things before now. And if you go back to, say, the 1970s, about a decade before Hubel and Wiesel won their Nobel Prize, von Fritsch, Lorenz, and Tinbergen received a Nobel Prize for developing the field of ethology, which is observing the action of animals, identifying species typical innate survival behaviors, and comparing the expression of those behaviors across species, understanding the stimuli that elicit those behaviors. So in ethology, a reductionist experiment looks something like this work that Tinbergen did, where instead of presenting very simplified stimuli like drifting gratings, you start with a stimulus that is ethologically relevant to the animal. So this is a herring gull chick, and if you ever go to the beach, you might see these seagulls with red spots on their bill. And herring gull chicks have evolved to peck at this red spot on their parent's beak to trigger the parent to regurgitate some food and feed them. And this is something that you'll see chicks and juvenile herring gulls doing, and then as they become adults, they'll stop performing this behavior. And Tinbergen showed that you didn't need an actual herring gull to elicit this behavior. If you took a herring gull chick and you just held out a cardboard cutout of a herring gull head with a spot on the beak, they would pick at that too. And if you held out a stick with some red spots on it, that they would pick at that too, but they wouldn't peck at just the head without the beak or just a head and beak without a red spot on it. So it's still a reductionist approach, but we've gone away from any arbitrary stimulus in the behaviors it's eliciting to something that is specifically chosen based on just kind of observing the animal and thinking about what it might be responding to. And a set of questions that Tinbergen developed in this area of ethology are dealing with understanding these kinds of survival behaviors, what they're doing for the animal and how they developed. So he posed these 4 classical questions of ethology. Why is an animal performing a behavior? What is it doing to increase that animal's fitness? How did the behavior evolve? Why is it that the Argus pheasant has decided to do this gigantic courtship display as a way of attracting a mate? And what is the balance between individuals that led to the development of this behavior? The causation of the behavior, and this is something that's more interesting from a neuroscience perspective. What is it that is eliciting the behavior that's performed? Is there a specific visual cue that's driving it? Is there a specific state of the animal that's driving it? And how does the behavior evolve over the lifespan of an individual? A lot of times you'll see animals change the way that they'll respond to their environment depending on how stressed out they are, how healthy they are, their age, their reproductive state. All of these things modify these behaviors, but the behaviors themselves are still this very genetically defined, very ancient thing that the brain has been developed to produce. So I'd like to talk a little bit about aggression because this is a form of survival behavior that I've spent a fair amount of time working on in my postdoc, and kind of look at some of the ways that people have tried to model this behavior in the past and then think about ways that we could do a better job of modeling it and understanding it in the future. And a little bit at the end, probably about some of the neuroscience of how we think that this behavior is controlled and how that relates to our theories of behavior control in the brain. So are there questions at this point before I dive in? Yep. So I'll start with the scene that opens Conrad Lorenz's book on aggression, which is that he goes out scuba diving in a reef in the Florida Keys and he sees a bunch of fish. And if you scuba dive for a while and you keep observing fish and you look at the way that they're interacting with other animals around them, you'll notice after a while that there's kind of 2 patterns of behavior across different species. You have some fish that swim around in schools, and you have some fish that are territorial and are always on their own and will drive off any fish of the same species as them. And it's these territorial fish that he kind of focused on for the rest of this introduction of the book. And there's this contrast. You have schooling fish, they all kind of have these shimmery kind of camouflagey appearances. They move in a mass together, and it's hard to kind of distinguish individuals from each other within this group. The territorial fish tend to have these really colorful displays. It's very easy to identify one of these guys and distinguish them from one of the other guys based on just coloration and appearance, and they're not trying to blend into their environment so much. So just from observing aggressive animals, thinking about aggression, scientists going all the way back to Darwin and going forward from there have thought about why you would want to have this mode of behavior in a species, why you would want to see some species that have very high levels of aggression that attack specifically members of their own species. You can have 2 aggressive species living alongside each other in a fish tank and they won't really care. It's only if there's another member of the same species that enters the tank that you'll start seeing aggression. And if you look at the behavior a little bit more, you'll notice that these attacks rarely are fatal to the animals. They're rarely leading to serious damage. And instead, what people have kind of arrived at as a conclusion is that aggression is a means of distributing resources and distributing population members. So aggression between members of a species helps distribute resources more to the members who are more fit, who are able to win aggressive encounters. It also helps to disperse the population. It acts as a kind of repelling force between different individual animals and spread them out more evenly across a territory. It can also inform sexual selection and help animals to choose the more successful mate. And there have been some really fascinating studies of aggression in social groups, in groups of baboons or crows that live in colonies where aggression is used to establish a social hierarchy. And building that hierarchy is a way of channeling the flow of information through the population of animals. So within groups of crows, you have senior crows that are more attended to when they generate alarm calls than more junior crows. So it's a way of propagating information among individuals in a group, and it's a way of sending signals and sharing knowledge between individuals. So there's been some fascinating theories of aggression that have developed over the years, mostly just based on watching animals in the wild and seeing what they do. And it really comes into these three different categories: aggression between members of the same species, aggression of predators towards prey, and maternal aggression. When an animal is protecting its young, it will become more aggressive than it otherwise would be. So it's not just random undirected violence, but it's this very specifically evolved thing that plays an important role towards improving the fitness of a species as a whole. So, yeah. Mhm. Yeah. Mhm. Yeah. I think that's mostly a measure of seniority and age, that the older animal is typically the one that's going to win the fight, and that's the one that is possibly more experienced and should be listened to. Whereas an alarm call from a juvenile might not be as informative because they haven't learned what's actually a threat and what you can ignore. But yeah, that's the area of aggression that I've done the least reading about, but is really fascinating to me. So if you go back and think about theories of aggression, a lot of them come from, not from physics, but from economics. Maynard Smith developed this hawk-dove problem as a classical model of aggression and how it can change or how it shapes the behavior of a species over time. And he did this, and he developed this famous hawk-dove problem, very closely related to the prisoner's dilemma, which describes when you might expect to see aggression in a species and when you might not expect to see it. So if you have hawks and or doves in a population competing over a resource like a bagel, then you can expect to see kind of two key strategies, or you can pose two key strategies that animals might choose to interact. The first is that you could take a hawkish strategy, which is that you fight with the other animal over the resource and you try to drive them off. Or you could take a dove strategy, which is that you don't fight over the resource, you just kind of have this slow, random decision of who takes the resource that isn't based on any physical effort. In a hawk strategy, there is a risk of injury in a conflict. In a dove strategy, it's just two animals that kind of have this posturing that takes more time to resolve itself, but it doesn't escalate to overt aggression. And so based on these two kind of ways of interacting with other animals, you can come up with this reward matrix for how things will resolve themselves depending on different pairings. So if you have a dove that encounters another dove, then around half the time, player one dove is going to receive the reward and half the time it's not. So you have a half of the total benefit of the reward goes to player one. If a dove encounters a hawk that's aggressive, the hawk is going to drive the dove off, so the dove is always going to lose and won't receive the food. On the other hand, if player one takes a hawkish position, an aggressive position, and decides to engage in conflict, they'll be able to drive the dove off, and so they'll get this big reward from having all access to the food if they come into conflict over food with a dove. But if they come into conflict with another hawk, then they're on average going to win half the time, and they have some additional likelihood of incurring injury when they're fighting. So there is a benefit that comes from the chance of them obtaining access to the food, but there's also a cost that comes from the risk of the animal being injured. So based on this framework of describing aggressive encounters, you can pose different scenarios where a hawkish strategy is going to win out or a dove strategy is going to win out. And to look at this, you can just describe the dynamics of these two populations over time. So the rate of change of hawks is going to be proportional, or here it's rate of change of the population, but really it's the net reward that comes to the hawks is going to be the expected reward from a hawk encounter plus the expected reward from a dove encounter. And the same scaled by the proportion of hawks and the proportion of doves in the population. The same for the doves. You have the expected reward when a dove encounters a hawk, scaled by the proportion of hawks in the population. The expected reward when you encounter another dove, scaled by your likelihood of encountering another dove strategy. And this led Maynard Smith to this idea of evolutionarily stable strategies, which is if you configure the parameters, the benefit of obtaining the food reward and the cost of engaging in conflict, you can formulate things such that a hawkish strategy will win out, or such that a pacifist strategy will win out just by manipulating the terms of this matrix. So this has been a popular topic in economics and in game theory dating back to 50s, 60s. And people have kind of iterated on these kinds of various simplified theories of aggressive behaviors and the way that they would resolve themselves over time to determine when a particular strategy is going to be advantageous to pursue. Just as another example, some people have devised this third category of behavior where whether you act as a hawk or a dove depends on whether you're in your home territory or not. And when you're in your home territory, you have an advantage. So yeah, there's lots of ways that you can kind of frame aggressive encounters between members of a species. And just by changing the costs and the benefits of engaging in aggression, you can come up with all sorts of different scenarios that favor a particular behavioral strategy just at the level of, do I escalate to outright attack when I encounter another animal, or do I just run away if it looks like he's going to beat me? So these are very old models, and they're things that people have kind of explored for a little bit, but what we really want to get to is not just this overall, on the level of a species, what strategy are you going to pursue, but what are you going to do within a particular aggressive encounter? And is there something strategic you can do at that level that's going to benefit you? And so what you'll see if you look at aggressive encounters across species from Drosophila up to white-tailed deer is that there is a temporal structure within a particular bout of within a particular instance of an aggressive encounter. And you'll see this kind of escalation of the intensity of aggression from low intensity, kind of sizing up the other individual, to more threat display-like behavior. I really like hard look as the name for a behavior, to more direct physical interactions, to all-out attack. And this is something you see, this is like a 1983 paper from Young Naturalist that was in some like Texas State Parks website on what to do if you encounter a deer. And this is Eric Hupfer's work from the Anderson lab looking at these same behaviors and the escalation of these behaviors in Drosophila. So in addition to deciding whether to attack or not to attack and deciding how intensely you commit to an attack within a species, there's this decision process that takes place within a single social encounter and this transitioning between different stages of behavior that takes place. And my slides are out of order, so let me find the next one that I wanted to show, which is that if you're interested in this, there's an excellent book, Dynamics of Aggression, that gets into some of these models. And basically one of the really cool results is that people have framed aggressive behaviors as a process of evidence integration. So you have some estimate of the relative fighting ability of two individuals and some degree of uncertainty in that estimate. And when you first encounter another individual, you're going to do something that is low risk, like a threat display, try to make yourself look big, and see if that gives you enough information to decide if you're going to win the fight or lose the fight. You'll do that for some amount of time, and then you'll transition to a slightly more intense form of aggressive display, maybe running past the other animal or charging towards them without physically encountering them. More energy expenditure, more risk, but gathers more information about how committed the other animal is to this encounter. And then only after you've gone through these stages and you're still uncertain about your relative fitness will you escalate to all-out aggressive behavior. So there's some fascinating work from people just like watching fiddler crabs on a beach and looking at the durations of these different stages of social encounters and showing that there is some evidence that animals spend a specific amount of time in one behavior state before they transition to the more intense and the more intense state. But all of this is pre-modern machine learning methods for quantifying animal behavior and trying to see if these things are actually present at the level of an individual encounter and how reliable they are. A lot of this is relied on still pretty controlled settings and on manual scorings of a set of behaviors in very nicely controlled conditions. And in practice, I mean, these are cool theories, they're interesting theories, and they make sense mathematically, but I think we still need to see how much they hold up in the laboratory and in what conditions they hold up, and to what extent this really is an information gathering process, and to what extent it doesn't seem to actually capture the behaviors that animals show in the lab. So what we've done is try to develop machine learning methods to improve our ability to quantify actions in social encounters, and actually go in and see, are these kinds of models a good way of describing what animals are doing when they interact with other animals? So you've seen plenty of pose estimation taking place so far. We put together our own system called MARS, the Mouse Action Recognition System, for tracking and behavior detection in interacting animals. Mostly developed for mice in the Anderson lab, but you can train your animal detector, pose estimator, and behavior classifiers in arbitrary settings. And we use these models to quantify animal behavior in high throughput screens and make sense of actions that animals are performing. And we started out just thinking we'll take a supervised approach, we'll define a bunch of different behaviors, and we'll use that to see what structure of behavior is present in these social interactions. Just to briefly go through the pipeline, we have a stage of detecting animals and estimating their poses, pretty much what you would get out of sleep or anything else. We got around the data limitation problem by crowdsourcing our pose estimations. We just put up 15,000 frames of mouse behavior videos on Amazon and paid people to annotate them, cost around $8,000. And from that, you can get out a decent data set of animal pose estimates and a good quantification of how accurate your pose estimation tool is compared to two different people trying to detect the same body part. So we can define our error in terms of the human rates of error. We then extract a set of handcrafted feature from those poses and train a set of classifiers to detect experimenter-defined behaviors of interest. And what I want to talk about from this project is mostly this question of how you define these behaviors and how reliable our definitions are, and think about what you can do with them and what else you could do to improve them. So for training Mars, we put together a data set of around 14 hours of manually annotated behavior videos that were scored for attack, mounting, and close investigation. About half the videos from male-male interactions, half from male-female. And we have this all released. We have training, validation, and test sets. You can go download them and use them for your own work. That's trained in a variety of experimental settings. And importantly, what we found is to get this to work well enough to be happy with it, we needed all of the annotations to come from one particular individual in the Anderson lab who didn't do this for this paper. We just took videos that she had annotated for other things in her own work. And the reason for this is that we got into this question of how people identify behaviors. And it's not something that you can do the same way that you can do it for pose estimation. You can't put up videos and have people on Amazon score them for you for 10 cents an image. But you can ask your lab mates to do it if they're stuck at home with nothing else to do. So we did a bit of a study of just how reasonable these human definitions of behavior are and how reliable people are in detecting a particular behavior like investigation or attack. And here's a sample of some of that data. You can download the data set as well and play with it. So it's just 10 videos, but just to get a sense of like, when we're making these ethograms of behavior and we're drawing these models of how you transition between different stages of behavior, how much subjectivity is taking place in describing behavior transitions. And yeah, if you zoom in on this, this is 8 different individuals within the Anderson lab. All of them were trained experts in behavior annotation with different degrees of motivation. I think I'll say these 2 at the end were technicians in the lab who were not depending on getting good results out of this annotation process. They were just paid to do it by the hour. And yeah, there are a variety of degrees to which people agree and disagree on the definitions of behavior. And I think this is going to be a big problem going forward if you want to compare experimental results between different labs that are ostensibly studying the neural correlates of the same action. So you have disagreeing on the exact timing of a behavior starting and stopping. You have some lumpers who put together all bouts of aggressive intent into a single annotation versus splitters who are very precise about when an animal is sniffing versus when they've moved their nose slightly away from the other animal. And yeah, you have periods where people just outright disagree as to what behavior an animal's performing. Because in a social aggressive encounter between mice, you'll have periods where an animal is looking for an opening to attack, but isn't outright attacking. Some people annotate that in terms of the animal's intent. Other people annotate it in terms of it looks more like they're sniffing than outright fighting. So if we're going to rely on these classical human-defined definitions of behavior, we have to be careful. Yeah.
How much instruction and what time to do
Yeah. Not super detailed. Annotator 1 trained annotator 3. Like who taught you how to annotate made a bit of a difference here in how you define things. Everybody had annotated videos before. I think we had one person here who had mostly annotated social encounters and flies, but everybody else did this for their own experiments within the lab. And I mean, yeah, like you can understand why some of these bouts are labeled as attack by some people and not others, because it is an aggressive intent without being outright attack. And it's more, I think this disagreement isn't reflecting some people paying more attention than others, maybe sometimes, but it's more just describing an animal's actions in terms of these two binary labels is not going to get us, is not satisfying for a person, and different people will draw different boundaries because this distinction, sniff versus attack, is a little bit arbitrary. Yeah. Mm-hmm. Yeah. Yeah, I think that's the direction that people are heading, is to either create a heuristic within the lab that defines it's an attack if they're moving at this speed and they're oriented in this way and the other animal's responding in this way. The other thing is to just come up with more than attack, not attack, but have like graded levels of attack. And we've done some attempts at doing that where we'd have people, I had a really fantastic student who was, I asked him to come up with different levels of aggression and come up with his own criteria for what divided them. Part of the issue is that the more fine-grained your behavior categories are, the longer it takes to divide them when you're manually annotating. This annotator 7 who put these massive bouts of behavior in finishes the job a lot faster than the people who are switching between individual labels. So ideally, we want something semi-computational, maybe with some domain-specific knowledge about what are the relevant features of movement that count as a behavior. And it's still a tricky thing to do, like how to go from pose estimation to a reasonable heuristic to define a behavior. We've worked with a group that has put together a set of heuristics to detect different kinds of social interaction in groups of mice. And when they like looked at videos and they would design these heuristics based on like looking at about a behavior and saying, okay, well, the speed is above this and the angle is this, and like try to define it that way. And when you went and compared the output of those heuristics to manual annotation, you would get F1 scores, kind of measure accuracy of like 0.1 to 0.4. It's 0.8. Yeah, I'll get into F1 scores in a little bit, but it's challenging. We really want some sort of machine learning method that's going to integrate some of this domain knowledge without relying on people setting these thresholds by hand. I put this in just to show this differences in the timing of bout onsets to further, yeah. It definitely depends on the question you have in mind. Mm-hmm. For sure, yeah. Like, do you want to know moment to moment how the animal is integrating sensory information to make a decision? Then your annotations moment to moment kind of matter. If it's just do the mice fight or do they not fight, then yeah, you can be a little bit noisy. And that's kind of been the challenge of this, is that there's not a one size fits all solution for these things, but it's going to depend on, do you need something that you can correlate to neural activity, or do you need something that's just going to get ballpark what's happening to give you the rough structure of a social encounter across individuals? Yeah. This is just to show that if you look at the timing, so what we did is we took an individual bout of attack or investigation defined as the median across annotators, or I think it was like at least 3 annotators labeled it as attack on, and we took the first, so yes, we took the median, we took at least 3 people label something as attack and define bouts by that criterion, and then we looked at the onset of attack and looked at how people labeled individual attack bout onsets relative to this group consensus. And you can see that people are fairly self-consistent. There's kind of a bias and a variance to these annotations. Some people consistently label start as happening the start of sniffing is happening a little bit earlier than others. Some people consistently a little bit later. And then some people are very precise in the timing of their annotations, and other people are a little bit noisier in when they mark the beginning of a behavior onset. So it's not just flat out noise. There is this self-consistency of behavior annotations that you get from different individuals. And we're thinking, could we use this as a way to get beyond attack versus sniff just by saying when different individuals are scoring things as attack, and use that as a different, and make that a definition of a type of attack? Like, is this bout, or is this period of time here where 3 people labeled it as sniffing a different type of attack versus this period of time here where they all agree it's fighting? So that might be one way to kind of combine the annotation styles of different individuals to get us to a slightly better resolution description of what animals are doing. Right, so now I'm actually going to define precision and recall. These are measures that you use for quantifying the accuracy of a classifier when you have imbalanced classes. Basically, your precision is telling you the ratio of true positives to true and false positives to detected positives. So an annotation that is present in a classifier but wasn't labeled in the ground truth is reducing your precision. Your recall is how many of the actual positive things you missed, how many false negatives you had. So if there's a bout of behavior and you miss something in the middle of it, that's reducing your recall. And to put these 2 things together, we come up with a number called the F1 score, which is just the harmonic mean of these 2 values. And that is something that is bounded between 0 and 1. And we tend to say an F1 score of around 0.8 is good enough to be usable. Individual humans have a self-consistent F1 score of around, well, yeah, I guess we'll get to that, around 0.9. So yeah, we will compute the F1 score for particular behaviors for different videos in this data set. Hang on, let me see what I'm showing here. So we defined one individual as the ground truth, and we just wanted to compute what is the F1 score, the average F1 score of the other individuals with respect to this ground truth. And what we got out of doing this analysis is we actually noticed that there isn't really a consistent F1 score for a particular behavior, but there's a really strong relationship between how long a bout of behavior is and what the average agreement between different annotators is. And so all these points are sniffing, attack, or mounting F1 scores from individual videos. And you see they all kind of lie along this curve. So in videos where all of your investigation durations are super short, you're never going to get really great agreement between different people that are annotating those bouts. Whereas if you have bouts of behaviors like mounting that last longer, you can get very good agreement between different individuals. So the duration of a bout is a pretty good measure for the accuracy of annotations, and this seems to mostly reflect this jitter in how people annotate the start and stop of a particular bout of behavior. Yeah? This is 30 hertz, sorry.
Yeah, right. This is in frames. Yeah, so there would be about a second. So, and this is all mouse behaviors in pairwise interactions. Yeah, so we then looked at the intra-individual F1 score versus the across-individual F1 score. So each of these 8 annotators scored 2 of the 10 videos twice, and there was a delay of about 8 months between those 2 bouts of annotation because that was submitting the manuscript versus submitting the revision. And these blue dots here are the F1 score of an individual versus themselves versus the gray dots are one individual versus the rest. And you can see that people are significantly more agreeing with their own definition than with other people's definitions. So this kind of suggests that we could take annotations from different individuals and train individual specific classifiers, and then maybe put those classifiers together to create a more fine-grained description of animal behavior. And without having to go in and very pickily, like manually define these different behavior categories, just kind of lump things together to create something that's more of a graded score of aggression or investigation as opposed to just this binary readout that we've been stuck with so far. Yes. So the classifiers that we were able to produce are about as accurate as another human in the lab. So this is the precision recall curve for one of our supervised classifiers that we trained based on human one's annotations. This is kind of the version of classifier that we took. You can kind of change your decision threshold to bias yourself more towards false positives versus false negatives. And you see that Mars kind of falls in among the other human annotators in terms of how close it is to matching the annotations of human one. So what I take this to mean is that if you have enough data, if you have, say, 14 hours of manually annotated training data from one individual, you can train a supervised classifier that's going to do a good enough job of matching how they define behaviors. It also kind of has the same pattern on a video to video basis that you see in humans, which is if you take videos with shorter bouts of behaviors where the mean human F1 score is lower, the mean F1 score of Mars is about the same. So it is still this like start and stop time of behavior bouts where these points of disagreement are going to emerge. And I think we can kind of use this as a way of deciding what is good enough for a particular behavior classifier. You can say that it's going to depend on how brief your behavior bouts are, and there's always going to be some jitter in their definitions. Yeah. Mm-hmm. Yeah, I guess it matters what you want to quantify for your assays. If you want to look at an escalation of aggressive behaviors, you have to look at attacking, but yeah. But I think this point here is kind of showing that you can classify mounting more accurately than, or not this point, but this guy back here. The attack points and the mounting points kind of fall along the same curve. So it's not that we can detect mounting more accurately than we can detect attack. It's that attack bouts are shorter overall, and that the shorter your bouts are, the less accuracy you're going to have. But maybe that's okay, because it's not going to be, you're not getting like egregious false positives or false negatives. It's just this jitter in the onset and offset of a particular behavior that seems to be the limiting factor in classifier performance. So it seems like we're kind of hitting this ceiling where you can match human level accuracy for annotating a particular behavior if you automate your system. If you take poses of interacting animals of decent quality and you extract features from those poses and then you feed those features into a classifier, you can do a pretty good job of detecting human defined behaviors. And the challenge I think going forward is going to be doing that more efficiently. So you heard from Talmo earlier about reducing the amount of training data you need for key point annotation. What we'd also want to do is reduce the amount of training data you need to detect a behavior of interest. And that amount of training data is really going to depend on the form of your classifier and the features that you're extracting from the animal's posture and feeding into it. For Mars, we just kind of fed it every single handcrafted feature we could think of. Angles between mice, velocities of mice, distances between all of the different pairs of key points. And what seems to help the most for making classifiers more efficient is just learning a better representation of the movements of animals in general. That helps us for summarizing what animals are doing and also helps us for training better behavior classifiers. So I'm not going to get into the details, but we have a method called Trebo that is aimed at learning efficient representations of animals' movements that seems to pretty significantly increase the accuracy of behavior classifiers. But the other approach you can take, which we have seen today, is you can try to just learn the behaviors of an animal in a purely data-driven way using representation learning. So you take the posture of your animal, you extract a set of temporal features from that animal's posture and how it's evolving over time. And then you use some sort of clustering method like t-SNE and watershed, as we see in Motion Mapper, to identify different dynamic motifs that the animal's showing in its movements. And Talmo showed some nice examples of this earlier on today, where you can detect different kinds of abdomen grooming, or I guess he didn't show, but things like wing extension or rearing in a mouse, just by looking at the dynamics of the posture of animals over time and trying to segment that into different categories of behavior in a purely data-driven way. And then instead of relying on human-defined behaviors, you can take these motifs and feed them into your downstream analysis. So is this a pipeline that folks here are familiar with? I kind of assumed that we'd have encountered Motion Mapper or MoSeq before. Maybe? There's a handful of them that have been developed, mostly for single animals. And it's all just kind of different approaches to taking postures of animals on individual frames of an experiment and looking at how those postures are evolving over time, and then finding recurring patterns, finding bouts of rearing that are happening over and over again, or bouts of turning just based on the way that key points are moving with respect to each other over time. So what we've done to try to contribute this is just to try to develop better ways to decide if methods are working or not. So you can take poses of animals and you can just do PCA on them and do k-means clustering, and you can, yeah? Uh-huh. It can be asymmetric, can be like one animal's approaching another or something. Let's see if I have. Yeah, I don't know if I have any good example slides in here. I should have given you a little more detail. Let's find that really quickly, sorry. One moment as I try to find a good example. Yep. Yeah. I have a few examples here. So the laboratory of Bob Dada developed one method called MoSeq, where you look at the depth of, you see that? Yeah. The depth along the spine of an animal, and you try to segment that into different patterns of how that depth is changing over time. And those patterns will correspond to different kinds of movements like walking or pausing or rearing. And in 2014 or so, Gordon Berman developed an approach like this in the lab of Josh Shavitz working with Bill on unsupervised behavior discovery in freely moving fruit flies. So you take a video of a fly walking in an open arena, you register it to a template, you extract a set of features out of the posture of the fly. Here it was multiplying this image with a line at different orientations to capture like the positioning of the legs and the wings, looking at the temporal features of those postural features, and then embedding that spectrotemporal representation into a low-dimensional space and performing clustering. So these kinds of methods give you different categories of movement. So periods when the animal's idle, periods where the animal's walking, where it's moving its forelimbs, and different individual clusters. Oops, let's see if these play. If you sample, no, it's not going to play. Well, that's anticlimactic. Exit and represent, I can try that. I'll have to exit both PowerPoints. It's okay. See if this works. Maybe? No. Sadness. It is really cool, I promise. Maybe it'll do it in here. Can you guys see that? No? Maybe you can see this one. Yeah. Aha. Is it not playing? Sorry, this is a background slide from another talk. And now my PowerPoint's crashing. Give me one moment to try to fix this. Technical difficulties. Okay, I think we're going to have to miss out on the videos because it doesn't want to play. But oh, you should look up the paper because it is pretty cool. Get back to where we were. Are we back? Terribly. Okay. Yeah, the screen has been dying. Is that one working? No, that one also terrible. What happened? Try it again. God. Okay. I am not sure. You think so? Can try that too. Can you see anything in this one? No. Okay. So there are lots of different data-driven ways that you could try to define behaviors, and people have put together a couple of these over the years. And what we've done so far in this space is just try to define a benchmark to evaluate the quality of an unsupervised embedding. So we, in the past year, released a data set called the Multi-Agent Behavior Challenge, and our goal is to develop a common framework for evaluating different unsupervised behavior evaluation methods. So we have a pair of data sets of tracking data from groups of interacting mice, 3 interacting mice or up to 10 interacting fruit flies with key points of the animals. And the goal is to look at the movements of the animals and characterize the actions that animals are performing. And using those characterized actions, see how well you can discriminate different experimental conditions. So for these 2 data sets, we have between 14 and 110 different sets of labels that might apply to any given segment of video and any given frame. We have manual or heuristically annotated behaviors of interest. We also have different experimental conditions, so animals that come from different experimental lines. We have periods of interaction during different times of day or different days in the arena. And we have different optogenetic manipulations of the interacting animals and different sexes of the flies. So we basically put together a whole bunch of different tasks. And in addition to these tasks, we have an evaluation pipeline. So given an unsupervised representation of behavior, it could be just the postures of the animal on a given frame. It could be the dynamics of that posture. We evaluate the quality of that unsupervised representation by passing it to a hidden set of evaluators that ask, given a learned representation of posture on a given frame of video, how well can I decode out different hidden labels about the task? So how well can I decode out when animals are interacting, the time of day, the strain of the animal, the sex of the animal, the optogenetic stimulation conditions, based on this learned representation of the original input sequence of pose data. So this is just wrapped up fairly recently, and we've been winning submissions from an online competition based on this. And the main thing that we're learning is that transformers are everywhere. Our winning submissions were mostly transformer-based deep neural network approaches that were doing the sequence-to-sequence mapping of animal pose trajectories. So our hope is that we can use this data set going forward to benchmark development of our own unsupervised methods. And rather than relying on human definitions of things like attack and investigation, we can try to learn behaviors of interest in a data-driven manner. And I think it's still unclear what approach is going to make the most sense to use. And it might, again, depend on the condition of your experiment and what it is you're trying to find out. In some cases, you really do still need human-defined labels to make sense of these behaviors of interest. And in other cases, it might be sufficient to just identify recurring motifs of animal movement and use those unbiased definitions of behavior as a means of describing what animals are doing. So that was just kind of an intermission on machine learning results. And now I want to get back to why we're doing these things and what we want to get out of these machine learning methods for studying animal behavior. Yeah. 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 I think the biggest thing is scale. You can take a classifier and you can run it on 10,000 hours of experimental data of mice given a bunch of different pharmaceuticals, mice at different stages of disease development, and you can quantify what's changing in their behavior across time without having to employ a lot of really miserable annotators to score these things out frame by frame. Also, you don't have to just use these discrete labels of action. You can extract out features of the animal's movement and describe it in a continuous space like you've done, and work in that continuous domain instead of relying on specific kinds of actions. I think for single animal stuff, there's an argument for both. For pairwise interactions, some of these social behaviors are just so complex that using a human definition of a behavior, I think, is still gaining you something over just a purely unsupervised and data-driven approach. But I think that's still kind of an area of development, is figuring out the right way to describe these social interactions. But the hope is that we can just scale things up and work with a greater temporal resolution, with a greater degree of precision and consistency than we could in the past. So yeah, that's kind of what you were asking, is why we're doing this and what we want to learn from all of these quantitative methods. I talked a little bit before about kind of old school models of social interaction and behavior. This escalation of aggressive encounters and what strategies for social interaction are evolutionarily stable. Going a step back from the individual actions that animals are performing during a social encounter, survival behaviors as a whole have interesting computational properties that should inform the neural circuits that are driving those behaviors. And some of these properties that have come out of really the more ethological observational studies of animal behavior are things like persistence of motivational states. So if you encounter a stimulus, your behavioral response to that stimulus should be able to outlast the stimulus itself. If you're a mouse wandering in the open and you encounter a hawk, even if that hawk flies off, you should be on alert for some period of time after that hawk flies off. As we saw in aggression, there's also an advantage to having a scalable behavior, to having a low degree of aggressive display versus a high degree of aggressive display for an encounter with a conspecific. And survival behaviors also need to be adaptable. So what this means is if you're hungry, you should be able to find food just by foraging, but you should also be able to learn that if you're feeling hungry, you can press a lever and get a food pellet. So the same motivational state of hunger should be something that you can hook up to both I don't know if there is a truly innate behavior, but to behaviors that you learned when you are very young, and you should also be able to hook them up to other learned behaviors. This is just to go through the example of scalability as a feature of behaviors. You should be able to escalate between different levels of aggressive intensity in a social encounter, and that should be able to move you up and down this, and you should be able to progress along this level of aggressive escalation until an encounter resolves itself, and you should be able to de-escalate. There's also some interesting work in what's called predator imminence theory on how your defensive behaviors should change in their form depending on the level of predatory threat that you encounter. So if a predator is just present in the environment, you might want to just be on alert and paying more attention to things around you. If you see a predator, you might want to freeze to avoid detection, but if the predator is coming towards you, you want to switch from freezing behavior to escape behavior. So two very different ways of reacting to a predator threat with a decision threshold between the two that really dramatically changes the way that you react to a stimulus. So survival behaviors have this kind of scalable and sequential structure to them in the abstract. And I also talked about adaptability. So an animal that's interacting with its world can encounter a variety of different sensory cues and can produce a variety of different resulting behaviors. So you might be water deprived, you might be dehydrated because of heat, an experimentalist might inject saline into you, and that can produce a bunch of different behavioral responses. You might drink more water when you encounter it, you might press a lever more times to get access to water, you might be more tolerant of something bitter in the water that you're drinking, depending on how motivated you are to obtain water. If you map these things all to all, it's pretty tricky to think of how you could generate a brain structure that's going to map any arbitrary sensory cue in very different modalities to any arbitrary learned or innate behavioral response. And it becomes even more difficult to modulate this mapping by other things like the presence of a predator or another animal in your environment. So one other kind of ethological observational concept that I think has a lot of importance for neuroscience and for understanding the structure of behavior is the existence of these low-dimensional intervening variables in between sensory perception and resulting behavior. Things like thirst and hunger and fear that are representing something that's kind of distilled away from the exact sensory cues that elicit them or the motor responses that you generate in response to them. And that can interact with each other at the level of motivational states as opposed to at the level of the sensory processing or the motor output. This idea has been around in ethology again since the 50s and 60s, where Tinbergen described this kind of high-level major instinct versus these low-level actions. And Lorenz has this awesome hydraulic model where your motivation to do something is a tank of water that's filling out, driving this behavioral response based on the amount of water pressure in the tank. But I think as we're able to observe more and more actions, we can probably do a better job of capturing the structure of behavioral control as you go from the underlying motivational state to the moment-to-moment actions that you're carrying out to produce that state. And also how you switch between different kinds of behavior programs that are driven by that motivational state. So when we talk about this transformation from sensory processing to behavior execution, this action selection thing is where all of these motivational state signals are really modifying the sensory motor transformation and changing the way an animal reacts in response to cues in its environment. So you can have different learned sensory associations, you can have different learned behaviors to receive access to a reward, but these internal states are something that's kind of isolated from those outside processing systems. And there's something that's more, Marcus Meister calls it the inner brain. There's something that's abstracted away from the exact processing of information that you're receiving from the world, or from the generation of motor commands that you're producing. And I think there's a real need in theoretical neuroscience to understand the control of these motivational states and how that's carried out in the brain and how they're informing this action selection process. Just to give a couple motivating examples, these things can be very kind of surprising and counterintuitive. An example that I really love from my collaborator, Muriel Zelikowski, is social isolation, which is something that people are more familiar with than they were in 2018 when this came out. So she studied groups of mice and the effect of being group housed versus single housed on animal behavior. And in animals that are socially isolated, they become more anxious. If you have a loud sound or a looming disc or a threatening cue, they'll freeze for longer than an animal that's group housed. And isolated mice are also more aggressive to other mice that they encounter. A mouse that's group housed with a bunch of other mice isn't going to go out picking fights with a new intruder. Isolated mice will run out and attack any intruder that's introduced to their home cage. And what Muriel found, which is really surprising, is that if you look at the brains of these animals, there's a neuropeptide called tachykinin 2 that is massively upregulated throughout the brain following a prolonged period of social isolation, and is causally associated with these changes in the animal's behavior. So she found that TAC2 is expressed in a variety of brain regions and becomes more widely expressed in social isolation. If you induce its expression in different brain regions of a group housed mouse, you can evoke just the aggressive phenotype of social isolation or just the anxiety phenotype of social isolation. And if you give the mouse a sonatant, which is a TAC2 antagonist, you can take a socially isolated mouse, dose it with this drug, and it will behave as if it's group housed. It will interact with other mice in a peaceful way. It won't show these stress-induced freezing responses. So when we think about how the brain carries out behavioral decision-making and how your motivational state changes the way you interact with your world, it's not necessarily going to be a problem of STDP and fire together, wire together, and Hebbian plasticity. This is a genetically wired change in neural activity, where you have a peptide that is upregulated when an animal is deprived of social cues from other animals. And the upregulation of this peptide is changing the dynamics of neural circuits in a way that changes animal behavior. And I think as we look at other kinds of experiences like stress or hunger, we're going to see more and more of these things where neuropeptides and neuromodulators are changing the dynamics of a neural population. And that change is what gives rise to a change in the structure of animal behavior when they encounter a conspecific or anything in their environment. We had another example from my postdoctoral work in the lab of David Anderson, where we found that mating experience dramatically changes the way that male mice interact with other male mice. So this was an experiment where we imaged in a brain region that's involved in control of aggression. And we found that if you took a mouse that had never mated before and you imaged the responses of neurons in this region to males and females, you got largely overlapping populations. But after an animal mates, if you look in a day later, you see largely overlapping populations of cells responding to male mice versus female mice. And as soon as you introduce a male mouse, these animals will run in and attack them. So this is, we think, a remapping of representations of conspecifics that's recruiting in a population of cells that's driving aggressive behavior in downstream motor controlled circuits. So we have this nice experiment where if you take a mouse and you give it mating experience and then you introduce a male at different latencies to mating, a non-mated animal is not going to show any aggression towards males, will show a little bit of mounting. If you introduce a male 2 minutes after they've mated, they show some mounting and no attack. If you do the same thing, but you wait half an hour between mating and introducing the male, you'll see a little bit more attack. And if you wait a day between mating and introducing of another male, you see this very robust aggression phenotype. even though the animal doesn't see males and females side by side during the mating experience. So it's not a activity dependent plasticity thing. It's probably some change. Well, it doesn't rely on seeing males and females side by side and learning to discriminate them. It's more likely to be some kind of change in gene expression in VMHVL that is changing the way that these cells respond to sensory cues from another animal. And here we know very little about what's actually happening biophysically to give rise to this change in behavior programs that animals are producing. So this is something that I think we're going to see more and more as we look at these kinds of survival behaviors. And I think the challenge as computational and theoretical people is coming up with models that can account for this kind of internal state dependent responding to your sensory environment. So let's see. And yeah, this was just another quote that I liked from Bateson back in the 80s. This idea of innate behavior is really a bit wishy-washy, and any species typical behavior that you're going to see is going to have some sort of sensory motor learning and also some sort of gating on based on experience. And I think a challenge in neuroscience is going to be making sense of the extent to which the brain is wired to produce a particular behavior versus the extent to which you need experience to generate those behaviors. So I've got, let's see, it's 20 minutes till 4. I had a more detailed dive into a particular study of aggressive motivation that I could do, or we could wrap it up there. Any preference? Yeah, any questions at this point? Mhm. Mhm. Oh, for sure, yeah. Yeah, I think things that are driven by like a spinal motor circuit really are innate, but if we get to things like defensive behaviors or feeding behavior, things where you're motivated to do them, but they're modified by your by motor learning and by the context you're in, it's trickier to really describe something as truly innate. Yeah, so like an animal is out foraging because it's hungry and then it encounters a predator. There has to be some mechanism in the brain where a high level motivational state that appears very quickly can override a low level state. And I don't think we have great models of how that happens in the brain. And I think that for the most part, we have people who have studied aggression and how it's controlled and people who've studied fear and how it's controlled, but there's less known about how one state overrides another. There is one really nice study from 2018 or so by Nick Bentley's lab looking at pain behavior. So in chronic pain, animals change they'll show like if you injure a paw, you're going to like spend less time out walking around on it and you'll want to go back to your nest, and you'll have like coping behaviors that you show like licking and paw guarding. And he found that in an animal model of chronic pain, a formalin injection, if you did this pain induction in a mouse that was hungry, you actually didn't see any of the licking and the coping behavior. And mechanistically, when he goes in and he looks at the brain region that seems to be involved in representing pain motivation, it gets shut down and it gets inhibited, and you lose the pain response in this region if a mouse is hungry. And the reason is that one region that's involved in hunger, AGRP neurons, release neuropeptide Y, and neuropeptide Y inhibits the activity of this pain region so that it doesn't even really activate if a mouse is in chronic pain and is also hungry. So the idea being that if you're injured, you should go back to your burrow and you should kind of take care of yourself and not go out running around in the world. But if you're in your burrow and you get hungry, your desire to kind of shelter yourself from the world shouldn't override your desire to go out and get food. So there seems to be this interaction between a hunger region and a pain region in the brain. such that the hunger region can inhibit the pain region and allow the animal to go out and forage when it becomes hungry enough. That's the best example I know of this kind of interaction between motivational states being linked to an interaction between brain areas. But I think as we go forward, we're going to start seeing more work that's uncovering how this kind of weighing of different motivations is carried out in the brain. Yeah. Yeah. To address that, yeah. Mhm. Yeah. No, all of this is, yeah, this is, it's a really interesting review on motivation by Kent Berridge. All of this is, you can make predictions out of this. So one would be if an animal has learned that it can press a lever for access to water when it's thirsty. If you then give the animal a saline injection and it has a mental model of this lever will relieve my thirst, it should learn the first time it encounters the saline injection, it should also be able to go and press the lever to alleviate thirst. So this is kind of like a model-based perspective that you don't have to learn a separate mapping from saline injection to press a lever for access to water if you have this intervening variable in between. It's quantifiable at that level, I suppose, or it's testable at that level, quantifiable, unclear. Also, whether thirst from a sodium injection is the same as thirst from eating dry food is not necessarily clear. And some things like Mhm. Yeah.
Is there something like that? So is it even the case, for example, that you know even if we have the same signal?
Yeah, there's been a little bit of work on that by shoot, I forget the guy's name. You can have different kinds of conflicts between motivations. You can have two appetitive choices and you have to pick between food or water. You can have an appetitive choice that also has an aversive stimulus near it. So if there's food, but there's something scary behind the food, you could have a conflict of, do I want to approach the food or do I want to run away from the threat? And people have posed things like that and shown that animals will show more hesitation if they have a conflict between appetitive and aversive cues than if it's just appetitive. And that the extent to which they're conflicted scales with the magnitude of the rewards and the magnitudes of the threats. Very coarse, though, quantitative, but not super detailed, more at this phenomenological level of describing the behavior.
The notion that we have this list of quotations.
Yeah.
Does it make sense to ask whether that can be included? Should I be able to quantify? I should be able to quantify that first year.
Yeah. Well, yeah. Mm-hmm.
But first year I get
yeah. I suppose you could measure the time an animal spends. Well, the way people will do this is they'll do like a progressive ratio lever pressing thing where you press a lever once, you get water, but then the next time you have to press it 3 times, and the next time you have to press it 5 times, and you can measure how far animals will go before they stop trying. Will they progress up to 9 lever presses, or will they progress up to 50 lever presses? So that's used a lot by people studying addictive behavior to look at how motivated an animal is for access to a drug if it's encountered it before, and how that motivation is changed by things like whether they're alone or if they're housed with another mouse. Very coarse, low-grained, low-resolution description of the animal behavior, but it's about what we have at this point. Yeah, so in that sense, yeah, there's a disconnect for sure between the machine learning part of my talk of we can detect attack and sniffing versus the, I guess, normative modeling part of the talk of what motivational signals are driving animal behavior and how do we quantify them. I think there's a need for relating the two to each other, for taking a more detailed description of animals' actions and improving our normative models, or looking to see if this like straight up interaction between intermediate states is really a good way of describing this behavioral decision-making process. Yeah. Mhm. Yeah. Yeah. I mean, I think we get better at a lot of innate behaviors with practice. Like a mouse that attacks another mouse for the first time isn't going to be as good as a mouse that's like a practiced fighter and knows how to charge in and attack. So even though the motivation to fight is innate, is present in all animals, the way that it's manifested as behavior is modified by learning. It's possibly the same with babbling, that there's a motivation to produce sounds, but then the sounds that you're making are part of a learned process. There's some interesting, I forget the name of the paper, there's a paper from the 80s where they introduced this concept of canalization, that you start out with the behavior being learned. And if it's a useful behavior, you then wire it in genetically and canalize it more and more and make it become more and more of a genetically driven thing. Very much a conceptual idea, unclear if that's really the case, but yeah, it's not a binary thing of it's innate or it's not innate, but it's more a question of to what extent is circuitry to produce this behavior kind of pre-laid out in the brain versus to what extent is it fully something that the animal learns. Yeah, the last bit I was going to show, I'm not going to have time to get into in detail. It was mostly looking at our efforts to study the control of behavior by the hypothalamus. People have shown that you can optogenetically elicit a variety of different survival behaviors like aggression and mating and social interaction and defensive fighting by stimulating different genetically defined cell populations in the hypothalamus. And as a postdoc, what I worked on was imaging from different brain regions in hypothalamus to see, or different nuclei of hypothalamus to see what they're doing as animals are running around and interacting with each other, and try to see, just looking at one nucleus at a time, what you could glean about the behavioral decision-making process that's taking place in these animals. And what I'll say is that the results you get from optogenetic stimulation and the results you get from imaging are pretty different. You can activate different genetically defined cell types and get like a robust attack behavior towards another mouse, or get a mating behavior, or get an obsessive following of three-dimensional objects. But then you image in these cells and you don't necessarily see cells that turn on when a human would annotate a bout of attack and turn off at the end of that bout of attack. And what I'll show that we've done recently is we've been using some dynamical systems modeling to extract out structure of neural activity in brain regions involved in control of survival behaviors. So in particular, we looked at a region involved in aggression called the ventrolateral portion of ventral medial hypothalamus, VMHVL. You can stimulate it optogenetically to elicit attack, but it has pretty much no neurons in it that are specifically on when an animal is fighting and off otherwise. But what we see is that when we fit a dynamical system to the neural activity, we find across all animals a single mode of the dynamics that has a really long time constant of integration. And if you project neural activity along that dimension, you get this gradual ramping up of neural activity over the course of tens to hundreds of seconds. The degree of escalation along this dimension is very tightly correlated with whether an animal is showing investigation, low intensity fighting, or high intensity fighting. We find individual neurons that contribute strongly to this dimension that have these ramping dynamics as well. And if you look at different individual animals, you can take the time constant of integration of this one dimension and plot it against how aggressive the animals are. And you see that you have some animals where this dimension is a very strong integrator. It receives input and remains persistently active for long periods of time, where the animals spend a lot of time attacking. We don't show it here, but the activity along this dimension remains elevated for tens of seconds, even in the absence of another mouse. And then we have other animals that never show aggression and show even their dimension of their neural activity with the longest time constant shows very short time scale dynamics and doesn't show this ramping behavior, and seems to be very tightly correlated across animals. So I'm skipping over a lot of the details here, but it seems like if you look in VMHVL neurons, you're not seeing this decision to attack or not attack, but instead you're seeing this gradual ramping up of an aggressive motivation that is very closely correlated with when the animal transitions. I think I have that here. When the animal transitions from investigation to dominance mounting to aggressive behavior. So some brain regions didn't look like this. Some regions you really do see a fine coding of the specific actions that animals are performing. But there do seem to be parts of the brain that have this more scalable, long time scale level of motivation looking signal. And I think a question going forward is, how is experience changing this time constant of integration? How are other external factors influencing it? And is this a common thing in other parts of the brain for other kinds of motivational states? So that's my abbreviated neuroscience data part. Yeah. Yeah, okay. I skipped all the details. We are fitting a switching linear dynamical system to the model. So you discretize neural activity space. You do dimensionality reduction with factor analysis. You get out an 8-dimensional summary of 150 neurons. You discretize that space into regions, and each region is fit with a linear dynamical system where your dynamics of your neural activity evolve according to a dynamics matrix A that is, yeah, and are allowed some external input just based on the distance between the animals. And the time constant is coming from the dynamics matrix for one of these dimensions of the, or one of these states of the fit system. Yes. Oh, okay. Yeah, like the science part. I thought you just didn't believe me. Yeah, so most regions subcortically are predominantly inhibitory. Like striatum is mostly GABAergic neurons. Most regions of the hypothalamus are predominantly GABAergic neurons. VMHVL is a ball of glutamatergic neurons, and we know that those neurons form recurrent synapses with each other, and VMH forms recurrent connections with another region involved in aggression, premammillary nucleus, which is also a ball of glutamatergic neurons. So it could be that this is reverberatory excitation between these regions, that they're getting driven with sensory input and that there's this gradual ramping up of activity over time. The other thing about these regions, like I alluded to with TAC2, is that they express like a dozen different neuropeptide and neuropeptide receptors. So it could be that this isn't ramping up of electrical activity, but it's accumulation of a neuropeptide in the region. These cells have a nitrous oxide receptor. They have a bunch of different opioid receptors. In addition to controlling aggression, these same cells have a big role in energy and metabolism. They send projections to the liver and they regulate insulin. So there's lots of weird stuff happening in here, and it's hard to know what exactly is driving this ramping, but we're working now on trying to knock down, the Anderson lab is working now on trying to knock down some of the peptides that could be involved in this ramping dynamics to see if we can get rid of persistence. Yeah. No worries. That's okay. We should, yeah. Yeah. It was about the same for each animal,
Okay.
but we captured like 8 or so PCs to capture 90% of the variance, and then we picked the number of states of the model that gave us the highest log likelihood.
I see. It is sort of like the states across.
All of the animals had this kind of progression of states that were correlated with investigation, low intensity fighting and high intensity fighting. Sometimes there was an extra state that was very rarely used for, like, there's a transient response when another mouse is introduced into the arena. But it's fairly consistent. Yep. Yeah, this is coming out soon. And just for comparison, here's state estimates from another region that doesn't have these ramping dynamics. This is medial preoptic area, which is involved in mating. And we don't get slow ramping dynamics, we get states that are kind of time locked with different stages of mating behavior. And trajectories in state space just kind of give you these periodic, these patterns of sequential activity of these neurons. So these are inhibitory neurons not involved in, and implicated in mating behavior, and don't seem to show any of these kinds of graded ramping signals that we pull out in VMH. I think since it's 4:02, I will go to a credit slide and thank. I showed you experimental data at the end. That was from Tomomi Karigo, who collected this data for another paper, and this data was reanalyzed by an awesome grad student, Adi Nair, who's been trying to characterize the dynamics of this region. And the computer vision stuff I showed you was from our collaborators at Caltech, Pietroprona and Yisong Yu, Christina Segalin and Jennifer Sun and Jelani Williams. And thank you guys for your time. We have other questions? Or yeah, we can also. So lots of physics in that one.
Yeah, it sounds more like, doing like the MAVE style approach of representation learning, but aligning it with the neural activity, getting like more bias rather than directed sense.
Yeah, our reviewer asked for that too, to see if we could find any unsupervised actions that were correlated with this ramping signal.
Yeah, the reasons you guys tried running the RSLDS on the behavior traces.
Oh, that we didn't do, no, to see if we could fit a dynamical system. No, we've just used it on neural activity so far. You could try some sort of joint modeling of neural activity and behavior, yeah. Yeah, haven't tried it so far. The model is getting the distance between animals and the facing angle, but those change on much faster timescales than 5 minutes that you're seeing here. Yeah. Yep. Mm-hmm. Like, is this activity what's driving aggressive behavior, or is it just kind of correlated with it? Oh, for sure. Reconcile that. Yeah, so if you stimulate this region optogenetically, at high intensity stimulation, you get aggression. If you ramp down the intensity of your laser, you actually get dominance mounting, which is this low intensity aggression I'm showing in here. We know it's not reproductive mounting because when they're doing reproductive mounting, they sing, and when they're doing dominance mounting, they don't. So it does seem like you can get a graded level of aggressive behavior by just stimulating this region in a graded manner. But when you stimulate this region, there isn't any persistence to the evoked behavior. So here, if you remove one mouse and introduce another, even like several minutes later, the resident mouse, if it's still kind of motivated to fight, will dive back in Versus optogenetically, if you stimulate and the mice start fighting, you turn off the laser, they kind of instantly stop and disengage. So there's something slow and persistent to the naturally occurring activity that isn't captured by the opto. When you elicit mounting? Yeah. Yeah. Mhm. Yeah. Right, yeah. So, yeah, so that's again, a shortcoming of the annotations is that we've been describing attack in terms of a human labeling attack, not attack, which isn't necessarily when the animal decides to attack. You definitely see mice that are motivated to fight before they actually start to fight. They'll do like tail rattling, they'll start moving around a bit faster, and that might be happening in this period here, and we're just not annotating for it. There ought to be some kind of behavior you could pull out in an unsupervised way, or just by looking at things like movement, bout duration, and speed that might scale with this signal in addition to just actually attacking. The other interesting question here is if you believe your annotation for the onset of attack, like on what time scale does the animal make the decision to attack? And can you tell based on how an animal's moving what it's going to do in the near future? This seems to suggest that the motivational state is linked with just whether the animal's in an attacking regime or not, but it doesn't predict the fine time scale of when the attack gets initiated. We don't really know where that decision is getting made. If it might not be happening in VMH. It could be that this aggressive motivation signal gets sent to a downstream motor region and integrated with visual input of like the other mouse is facing away from me and it's time to dive in, that actually drives the attack bout itself. And Anna Gert-Faulkner has some work that kind of suggests that as well. Mm-hmm. If you get a transition from between behaviors? Yeah. Mm-hmm. If it would make it. Is needed for the persistence. I don't think we've tried that ramping specifically. There's a neighboring part of VMH DM for dorsomedial that is involved in defensive behaviors. And there, if you stimulate it, first of all, the kind of defensive behavior you get scales with the intensity of stimulation. You get freezing or like attending versus freezing versus escape behavior, depending on how intense your stimulation is. And I think there is a little bit of persistence to the escape behavior after the stimulation turns off. Any other questions? Yeah, thank you guys. Yeah, thank you so much.